Bonjour et bienvenue sur cette première session intitulée Biologie du développement, cellules souches et cancer pédiatrique. Je me présente, Élise QUILLENT, je suis médecin et responsable dans le bureau du conseil scientifique de la fédération Grandir Sans Cancer et je commodère aujourd'hui cette session avec Celio de l'Institut Curie que je salue.
Bonjour à tous, donc je suis Celio, je dirige une équipe de recherche qui travaille sur une tumeur pédiatrique qui est le méduloblastome à l'Institut Curie. So I'm going to switch now in English. First, I wish to ask all the speakers to keep their time since we are in Visio. It's very, very important that they keep their time. So you can ask many questions as you want on the chat, and we are going to discuss all these things after the talk, directly after the talk. So now I'm going to introduce Richard Gilbertson. It's a real pleasure to have Richard with us. Richard is the director of the CRUCK in Cambridge. CRUCK stands for Cancer Center UK, and is also the director of the oncology department. He's doing very, very original basic research as well as translational research. And his research focuses on the link between normal development and the origins of childhood brain tumor, in particular medulloblastoma and ependymoma. He used very relevant and faithful model using mouse genetics and primary cells, and he have made very, very important achievement in this field. So now I'm letting the stage to Richard.
Great, thank you very much. It's nice to be here. Can you hear me okay? Great.
Yeah, it's okay.
Okay, so, and hopefully you can see my slides. So what I'd like to talk to you about today is the origins of childhood cancer and how we might use that to prevent adult cancer. But I'd like to thank very much the organizers for this opportunity to talk to you today. So if we think about this, the first thing I would like to talk about is some of the big challenges that we often brush over in pediatric cancer. Across the field of oncology, we have some major issues facing us. This is somewhat out of date now, but the numbers tell the story, and that the numbers of patients with cancer over the next 3 decades and the cost of that globally is going to dramatically increase. Another important concern is that about 1/4 of all malignancies in the developed world are diagnosed in the emergency room, accident and emergency department, and most of those patients do very badly. Also in pediatric cancer, some of the diseases that I'm interested in, because of genomic advances in particular, we've divided those patients into very small subgroups of disease. So in the country where I work, in the UK, some of the diseases only have 2 patients available for a clinical trial each year. Clearly that's untenable. We know the story of drug development, only about 1 in 10,000 compounds that even make it to a druggable form will ever get to the clinic. And then in childhood cancer, sadly, children who are diagnosed with cancer in Africa are about 5 times more likely to die of the disease than those in developed countries. So clearly this is unworkable and unsustainable. We have to have a new way of tackling cancer as we move forward. But there is a big opportunity. This graph shows average survival, life expectancy of just the population going back over the last 200 years or so. And apart from the blip with Spanish flu, there's a dramatic increase and steady increase in survival and life expectancy of the population. And that has all been achieved through primary prevention. However, in cancer, we continue to focus at the end of the disease. Many of our patients are diagnosed with metastatic disease when the disease is rampant and incredibly difficult to manage. And we've ignored the earlier steps in the disease and really the cells in which this disease develops. So what I'd like to focus on is, where does cancer come from, and how might we use that to prevent this disease from ever happening in the first place, rather than just focusing on this aspect of the disease. So I'm going to skip over this for time because it's work that we've published over many years, but we began our journey in this question when I first worked at St. Jude about 20 years ago now, and if Charles Mulligan's are listening, I don't think either of us would believe it's that long ago. But we started by looking at neural stem cell types in the mouse because we were interested in where brain tumors were born in the nervous system, particularly those in children. And we looked at a number of different stem cell types, both topographically and temporally. And what was surprising is that using old-fashioned affymetric arrays, when we compared the gene expression profiles of these cells with tumor types, actually they were remarkably similar. And it allowed us to map the origins of tumors in the brain in children to those same cell types in the nervous system, because they carried forward this transcription a little bit like a molecular address. So for example, Paul Gibson, working in my lab, was able to show that Wnt medullos, previously thought to arise in the cerebellum, actually arose on the dorsal brainstem and grew up into the cerebellum, recapitulating very much what we see in patients on this MRI scan. They're very different from hedgehog tumors, which arise from granular neural precursor cells and are confined to the cerebellum. And we've done large MRI studies now, as have many groups around the world, to demonstrate that this developmental signature is true, and that Wnt medullos are all really arising off the dorsal brainstem from mossy fiber precursors of the lower rombate lip, distinguishing them from hedgehog tumors, which are largely confined to the cerebellum. Now, we've gone on and used this approach to look at many tumor types in the nervous system. We focus particularly on the ependymomas, which occur throughout the nervous system, showing those arise from radial glial populations in the region of the nervous system where you find those tumors, and choroid plexus tumors arise from the initial cells which form the choroid plexus, and we've published extensively on this. But really, all we've done is validate the incredible work of Bailey and Cushing, who were working over 100 years ago in the northeast of the United States, and armed with nothing more than a microscope and a few simple stains. They predicted all of this, which cost us a lot more money, I can tell you, to demonstrate. So just using morphological characteristics, and often sort of envisaging the certain cell types that might exist, that we've gone on and demonstrated exist, they actually sort of in a prophetic way said that these tumors must arise within the context of development. So that leads me on to sort of the next part of my talk, because we're going to hear a lot about children's cancer today, but I really want to think about cancer in its broadest terms as a disease. So in children, it's very easy for us to consider cancer being a developmental disorder. We see that because most of the solid tissues are blastomas. They're sort of extensions of embryonic tissue. However, in adults, if we look at the patterns of cancer that develop, they're very prescribed. They occur in the same incidence, in the same times during development in adults. And essentially, many of us believe, and it's sort of largely accepted now, that adult cancers, while of course provoked and induced by toxins in the environment, have their roots in development in a similar way that pediatric cancers do. So I'm going to share with you briefly some work that I did when I was still at St. Jude, before getting on mainly to talk about unpublished work. And this was a collaboration with Arza Thomas, who still works at St. Jude as a fantastic mathematician, and Lee Chin Zhou, who was a postdoc in my lab, now has their own lab at St. Jude. And again, we've published this, so I'm just going to go over the principles of it for time. But essentially what we were interested in is understanding where cancers come from. What is the ground state that is required to produce a malignant disease? And we were interested, because of that concept I've discussed, looking at this both in neonates at postnatal day 1, but also in adults. And so the first thing we did is using a Curie mouse that Lee Chin made in my lab, which basically had Curie RT2 knocked into the PROM1 locus, the CD133 locus. We lineage traced multiple organs and asked the question, in which organs does this Curie actually label stem cells or fully differentiated post-mitotic cells? And then we could look with genomic technologies at the gene expression patterns of these, as well as measure their proliferative capacity. And to cut a long story short, basically we saw lots of different patterns of these. So for example, in the small intestine, this is neonatal recombination, this is the P60 combination, immediately after day 3, and then after a long term of lineage tracing. You could see in the stomach, small intestine, liver, actually at P3, these are remarkably regenerative cells. For example, the very rare population of 0.001% of cells around the portal system give rise to the entire liver within about 3 months of birth. However, these cells become quiescent in adulthood. They just sit there unless the organ is damaged. So you're able to measure the relative lifelong generative capacity of these cell types. And we could go ahead and look at the transcription of these and the degree to which they mimicked stem cell signatures that you find in sort of archetypal stem cell populations. Now, having defined what the lifelong function of these cells is across 12 organs, we then turned our attention to what is the malignant potential of these cells. So we know what their normal biology is. Now, what happens if at the same time points, using the same Curie at P1 or at P60, we now introduced oncogenes through Curie recombination or deleted tumor suppressor genes? So what Li Qin did then was to follow these mice for the duration of their lives, and these are the survival curves of these animals, involving about 800-900 mice, and doing autopsies on all these animals, and looking for the presence of tumors in mice that carried a set of lineage tracing alleles, as well as inducible oncogene and tumor suppressor alleles that covered the waterfront of the type of oncogenic mutations you see in human cancer. Now, because we were able to look at so many mice and we did autopsies on these animals, we could generate heat maps of the incidence of where these cancers occurred. And what we did then was to integrate this data, because we knew, for example, what their proliferative capacity was, we know what their normal differentiation capacity was, and we also knew what their lifelong lineage tracing capacity was. Because we'd also, on the other hand, measured the tumor forming capacity of these cells, Arzu could invent this new mathematics, which took into account all of the observations across these many hundreds of animals, asked what's the relationship between, for example, the lifelong generative capacity of a cell, a particular PROM1 cell's ability to make other cells in that organ, and also the risk of that cell making cancer. And what you're seeing in the graph here is all the data for neonatal inductions in pink and for adults in green. Now, what was interesting is if you take any lifelong generative capacity, let's say we have a PROM1 cell that has the ability to make about a quarter of the organ, then the risk of that cell making cancer is about 10% throughout the life of that animal. However, in adults, it's much higher. It's about 7-fold higher. Now, this isn't an issue about adults taking longer to accrue mutations or having that ability to accrue mutations, because remember, the neonatal cells were induced at P3. So these cells have carried mutations throughout the life of the animal, and yet they're still protected relatively from cancer risk, compared to those cells that were induced 57 days later, which is a long lifetime, a long span in the life of a mouse. So this provides the first evidence that perhaps neonatal or juvenile stem cells have a protective mechanism that prevents them from getting cancer relative to their adult counterparts. So this gets to the sort of unpublished aspect of the talk, which I'd like to spend much more time on. And it also helps us think about a question that I've really been interested in for many years, which is perhaps odd for a pediatric oncologist, is could this explain why children don't get that much cancer? So this is a graph on the left axis, the growth velocity of humans. So obviously at their highest and peak in utero, falls during the first decade of life as a peak in adolescence, and then drops again. And this is the risk of developing cancer, the number of cancer cases per 100,000. Now, of course, it's much more common cancer in adults, and the dogma is that that's because it takes this long to accrue mutations, kind of environmental insults like smoking, alcohol, UV exposure, etc., to accrue enough mutations to be able to generate malignant disease. Now, that's almost certainly part of the story. However, it ignores the fact that nonetheless, children undergo enormous mitotic and differentiation stress during that early critical period of development. And yet cancer is remarkably rare, although we do see it in certain children. And there's a long-standing theory which has existed, known as the cancer protection theory, which is that there must be an evolutionary mechanism that is developed which protects multi-organ species as they move from lower order species to becoming multi-organ species, that has protected this critical period in development from tissues becoming malignant. So as organs are rapidly developing, they're undergoing huge mitotic and differentiation stress, and they're encountering toxins in the environment for the first time. Are these actually resulting in insults that the organism has evolved a mechanism to protect it from cancer? And of course, you wouldn't need this later in development because, of course, you've already passed your genes on and you just need to repair tissues rather than prevent cancer. And if we could understand that, could we prevent cancer in adults? So this is where it gets a bit bonkers. So this is the work of Amir Hasim, who's a postdoc in my lab. And Amir took this observation, and now what we've done is take those same multiple organs that we mentioned that Li Chin had looked at, and we've isolated those same populations from those mice throughout development, from embryogenesis through to late adulthood. And we've generated, in collaboration with Rudy Abersol, Wolf Reich, and Shankar Balasubramanian, both the proteomics RNA transcriptome and the methylome for these same cell populations. Now, the idea here is if we have that information, we can now map this exquisite detail of the genomes, of the genome, proteome, and methylome onto these functional aspects that we've measured, lifelong function and cancer risk, and try to get to understand what the biology is that's controlling these various issues. So this is just a snapshot of some of the data. There's about 800 individual samples of PROM1 cells here. It doesn't look like that many because, not surprisingly, based on organ, they're all tightly clustered. It's colored here based on organ. However, what's quite remarkable is this is not the heterogeneous population of cells which comprise an organ. But remember, these are just those tiny fractions of PROM1 cells that we've isolated from that. But nonetheless, they have organ-specific transcriptomes. Now, on top of that, we have a huge amount of data. We've got their cancer risk, we've got the sex of the animals, we've got the time at which induction happened, we've got methylome, proteome, transcriptome data, and also we've developed damage data. And this is truly a big data problem, more than we were able to manage with conventional analyses. So we've turned to deep learning, and particularly neural networks, to try and solve this. And I'm just going to give you a brief overview of what a neural network is for those who aren't familiar. And essentially, this is an in silico system by which you can introduce a series of samples which have associated information with them, such as the sex of a sample, the age, or the organ of origin. And that data, in this case, it could be transcriptional data, will flow through the neural network and encounter a series of layers, hidden layers, at which there are nodes or neurons. And at that point, essentially the algorithm will decide whether or not this particular piece of information contributes to the understanding of that input data. In other words, it helps me understand that this was female, or it helps me understand that this was a cancer risk. And then your information is output in a form which allows you then to interpret that. And then you can put unseen information in and continue that work through the neural network. So just to illustrate that, this shows how we've trained a neural network based on organ type. So for example, we've put in a training set here and told the neural network that some brain, kidney, small intestine, large intestine, etc., the tissue of origin of these particular cell types. And what you're looking at here in this confusion matrix is the capacity of that matrix or of that neural network to then see an unseen cell type and predict where it came from. And it's remarkably accurate at doing that. Now, again, remember, these are just a small fraction of the stem cells. So it's quite interesting that a small fraction of cells, for example, in the liver, that just around the portal system, nonetheless, using the small information that comes from that population, the neural network is able to identify that as a liver. Perhaps even more interesting is it's able to do that based on age. So we can train the network based on across all cell types, what is it, P3, P15, P180, or almost 2-year-old mouse. And again, the system will do that. It will take a cell type which is unseen and very accurately predict what age that cell type is. Now, when you delve into the neural network for a sense check, you start to see the kind of genes that you would expect the decision is being made by. So, for example, in lungs, there's a series of genes which you'd expect to predict lung tissue, liver tissue, or prostate, like, for example, NKX3.1, which is or probacin, which are well-known prostate-associated genes. So it tells you that actually the neural network is actually making decent decisions based on known biology. The real exciting part of this, however, is what about unknown biology? And that's when we bring all of this together. So what about cancer risk? So these letters here along the bottom are really classes that we've developed in our system to define cancer risk. But what's really exciting is we can take a set of these stem-like cells that we know they're at lifelong risk of making cancer, and we can train the neural network on what transcriptional signature is associated with a particular cancer risk. Interestingly, then, if we put an unknown cell type in, it will very accurately predict what the cancer risk is of that cell type. Now, you can imagine the biological implications and diagnostic implications for this are tremendously exciting. But how do we explore this on a sort of global organism-wide scale? And this is what we call a universe plot, which is a particularly exciting way of looking at the data. So what we do is we take the Pearson correlation efficiency, which is a metric of association between a particular gene and the way that the neural network is trained around a particular observation. And it starts to self-organize gene expression around the training that the neural network has gone through. Now that sounds like a sort of lot of gobbledygook, but when I show you this next example, you'll understand why that's very powerful. So we put these genes into the neural network in a completely agnostic way. We do not tell the neural network what the biology is of each of these genes. However, this large purple cluster in the middle here has self-organized through the neural network the entire machinery which is involved in the cell cycle. So what that tells you is that by using this entire clustering of data and then putting that into the system, it is able to recognize known and real biology. Now, what's tremendously exciting about that across these 12 organs is that many of these clusters, which are as tightly organized as the cell cycle, currently have no known biology associated with them. It's not understood why these genes would co-cluster. Now, of course, it may be artifactual, or it may just be due to our system, but it is also highly likely that there is real biology in these. So when we extend this into the entire network of cells that we've produced, so we have now a system of about 1,500 individual samples across all these organs, across all stages of development, you can start to see this universe of aging where we see connectivity between individual genes during the life of the animal. And if you like, getting the first view of the holistic transcriptomic changes which happen as cells go through these processes. And you can look into this and ask the question, what are relationships associated with genes which are specifically important for cancer risk in particular organ types? So we've extended this now to a multi-deep learning approach to integrate across many, many tissue types. So we've done this for single cell sequencing, bisulfite sequencing, and also now in damage. So in the damage approach, what this allows us to do is to take individual cell types, and we've done this for the lung, we've done it for the large bowel, and we've also done it for the liver, is to actually not just look at normal development. So what happens to these patterns and the neural network during normal development, but what happens to the neural network if we damage that organ in a cell population that's normally quiescent. So it allows us to wake these cells up and then look at these cells through this neural network. And what's remarkable in the context of damage is that these cells are now predicted to be extremely young. So in, for example, an adult liver, which might be a P300, if we damage that liver, allow that liver to repair and remove the stem cell population that's repairing the liver, those cells don't look like a population of cells that are in an adult at P300. They actually look like a cell at P15, so very early postnatal life. That's also true in the large bowel if we damage that with DSS. It's also true in the lung, which is damaged with another chemical damaging agent. So it sort of gives you this concept that following damage, there's truly a sort of waking up of these cell types, and they're then going on to repair the lung or in the liver or in the large bowel in a way that's different from the adult population. Now, I don't have a chance to show you because of time, but actually, if you look at those cell populations, you do see the signature which allows you to define this as a P3 or a P15 cell. But there are large swathes of the biology missing. So that's the point of those large sort of universe plots. We start to see populations of genes that are actually absent, which would normally be present at P3. And we think those are the populations of genes, the cohorts of genes, which would be protective in a neonate and stop cancer from happening in the first place. So that's what we're excited about delving into next. So I think this is my last slide. So what's the idea? So in babies, every single organ we know has a stem cell population or a progenitor population that is responsible both for self-renewing, but also for generating the multiple populations of fully differentiated post-mitotic cells that populate that organ. Now we know that neonates, humans as well as other higher order species, are insulted through mitosis, differentiation, and other environmental stresses. But we believe through the work that Li Qin's shown that there's this ability to protect the organ from those and resist it, to stop them resulting in the sorts of stresses which result in malignant disease. So the organs can go on and produce not scarred, not malignant tissue, but normal tissue, so that organism then can live long enough to propagate genes, etc., and pass on through sexual reproduction. In adults, however, there are stem cells existing in adults. We know that they're largely quiescent. And so they're sort of not active at any one time, like those cells I showed in the liver. However, in response to environmental insults, rather than actually have to repair in a perfect sense the organ, they just have to plug the gap, and scar tissue is the classic example by which that happens. However, because they don't need to protect against lifelong malignancy risk, there is an increased possibility of those cells producing malignancy as much as repairing the tissue. What we'd like to do with the kinds of approaches we've shown you is identify what is that mechanism and could we reactivate it in neonates? Perhaps it's an epigenetic mechanism, or it could be a translational mechanism, and that would allow us to reactivate that in adults so that the cells in adults don't just look like P15, they completely behave like P15, reducing the risk of malignancy in those cells. So that's my last slide. I'd like to thank everybody in the lab who's done the work, particularly Amir, who's done most of the original work that I demonstrated at the start. And thank you very much for your attention. I look forward to any questions.
Okay, thank you, Richard, for this very nice talk. So we are now going to go through questions. So I encourage all of you to ask questions on the chat. On top of that, if you don't want any translation, you just click on the flag on the English flag on the website, and you will not have the translation below. Sorry? Okay, so Richard?
Yes.
Yeah, okay. So there is a very, very general question from the audience, from the chat, asking if pediatric ependymoma can be cured. It's not completely related to your talk, but it's like a general question. So.
Yeah, so I think that we very much hope the answer is yes, and I believe the answer will be yes. I think what I can say is that the understanding that we've developed of ependymoma in the last 20 years has dramatically changed the way we view the disease. And so what we do know now is some of the fundamental genetic changes which are responsible for driving that disease. I think what's been quite encouraging is that those fundamental changes, rather than being necessarily complicated, are quite simple, and you often need just one change to drive an ependymoma, whereas some of the cancers we see in adults, for example, there are a myriad numbers of mutations which are responsible for those cancer types. So that allows us to have specific targets. I think the other encouraging aspect is one of the main drivers we found, for example, in a form of ependymoma in the forebrain. We understand a lot now about how it causes ependymoma by corrupting the normal processes which activate genes. And in collaborators around the world, particularly a good friend who works at UCSF, we were able to develop techniques which block that from happening, so prevent this molecular switch from binding to genes and activating the genes that it shouldn't normally be switching on. So it's switching on light switches, if you like, all around the genome, and that it shouldn't normally be doing. And so this drug seems to be able to stop that from happening. And in fact, this may well be important for a number of genes, and my colleague and friend Charles Mulligan, who'll be talking in this session, looking at very similar approaches in leukemias. So I think, this is also gives us encouragement that not only will it be true for ependymoma, but we'll be looking at these kinds of approaches together, which will further accelerate it. So I think, yes, there's lots of hope at the moment in terms of the understanding of ependymomas and new ways to treat it.
Okay, so maybe to initiate the discussion, I will start with one question. So you mentioned that pediatric stem cells could be more protected from the cancer arising. So my question is, how do you reconcile that pediatric tumor need less mutation than adult tumor if they are like less susceptible to or less prone to cancer? And what is the mechanism that could molecularly protect these pediatric stem cells from being transformed into cancer cells?
Yeah, so that's a great question. So I think it actually gives us clues as to what that might be. So I mean, certainly not all cancers in children have few mutations, as you know. Some of the brain tumors have very large numbers of mutations. So it's not, and as do some of the solid tumors in the peripheral organs. But there are remarkable tumor types like retinoblastoma, for example, where you can perhaps just have a mutation in the retinoblastoma gene, which is really interesting. And ependymoma, as I just mentioned, we've shown that just the fusion on its own can drive the disease. I think what we're understanding is that there are fundamental aspects of immature tissues that are able to go on and drive the normal processes that embryonic tissues do. So embryonic tissues are set up to grow, fill an organ, and then basically stop and differentiate. I think what that suggests, and there's colleagues of mine in Australia who are working on this at the moment, where we think actually what might be going wrong is that switch to actually prevent differentiation from happening, the maturing of the organ. So it takes the focus off the sort of classic oncogene, which is a driver of proliferation, or the tumor suppressor, which is a block on growth or death, to something to do with differentiation. And so what it might be is a mutation, a second event in a gene, which normally would be responsible for activating differentiation. Or it could be the way that the genome is remodeled from a methylation perspective, which perpetuates the sort of embryonic state rather than the mature state. So I think what it actually gives us is a clue as what that process might be, as much as sort of, rather than saying, well, how does it, a single gene can actually drive that disease?
You have one question, Didier? Okay.
Thank you very much for this really impressive talk. I have actually a question. With your deep learning methods, do you understand me? Yes?
Sorry, I can't hear the question.
So is this microphone on?
I can hear you, I can't hear the other one.
Yeah.
Can you hear me? Okay, maybe.
Yes, yeah.
Yes, it's okay now. Okay, go on, Didier.
So thank you very much for this really fascinating talk. And I was wondering, actually, using your deep learning methods, you have a way to measure the aging of your different population. And actually, yes, would this aging be different between the different tissues, or is it a universal aging?
Yeah, that's a great question. So actually, we don't. So what's quite remarkable about this is, although the cells are taken from different organs, it is organ agnostic. So although the deep learning approach can actually identify organ type if we train for organ type, so training for liver, training for lung. When you train for age, that's organ agnostic. So we simply put all of the cell types in, regardless of where they're from, and say this is P3 and this is P16. So what it allows us to do is identify a core signature which is associated with being a particular age, irregardless of where that cell comes from. So there are core signatures associated with aging in these cell types, as you're sort of alluding to, which aren't dependent upon the site that that cell came from.
And maybe if I can, just a second question. For instance, you also have heart tissue in this analysis, and we know that basically heart never developed cancer, right? Was that an outlier in your analysis? Or, I mean, can you explain why some tissue are more prone to get cancer than others with your analysis?
Yeah, so we don't have heart, unfortunately, in our organ system. So the other limitation of this approach is obviously because it's such a massive study, and the way we've done this is use the PROM1 allele, because that's where we did the lifelong lineage tracing and the cancer risk on. So we're restricted to that population of cells. We used it because in some organs, they're post-mitotic differentiated cells. In other organs, it marks stem progenitor cell populations. So it allows us to look at cells with different capacity using a single Curie allele. But of course, there may well be cell types, and there probably are, I'm sure, in the heart that have that capacity, but they're not labeled by prominent one. So one of the limitations is that we're restricted to where we see particular patterns of prominent one expression.
Okay, we may have time for a last question. Go on, Thomas.
Yes, fantastic.
Can you switch on this microphone?
Fantastic talk. Do you think that, or do you see already in your data that in the different type of tissue you have common molecular nodes that would be expected to be commonly targeted in all pediatric cancer, for example, or do you expect, in fact, that as the tissue are very different and cell context is very important, the different nodes, important nodes, will be quite different from one cancer to another?
Yeah, that's a great question, thanks. So actually what's surprising, perhaps it's not surprising, is so if you were to invent a human from the start, you probably wouldn't invent fundamental systems that were different in each organ because it would be unnecessarily complex. So the hypothesis that we began with is while organs may differentiate differently in terms of the way they express themselves in post-mitotic differentiated cells, the fundamental mechanisms that control important aspects of the cell biology are probably common in different stem cells. And that's somewhat borne out by the fact that, for example, cancer risk is organ agnostic. And as we talked earlier on, age is organ agnostic. And I think that's giving us clues that actually some fundamental aspects of the biology of these populations are common across organ types. Now that gives us hope that as we look for these mechanisms that prevent cancer, because they're likely to be fundamental, they're probably shared across organs. And so we've already got some, I mean, we've just started to collect all the data from the damage. So the key aspect here is, okay, we've got normality now. We know that damage increases cancer risk. We've shown that in our mouse models. What is it about the transcriptome in a damaged organ that's different from that of a normal adult organ and different that from a child neonatal organ? And that's where we're going to start to see these differences in the universe, where we can look in and dive into those nodes which are somehow absent during damage response. But actually I can tell you that just looking across tissues as diverse as the large bowel, lung, and liver, there's absolute commonality that we see, both in the damage response, but also in what's different between an adult and a neonatal cell. So I do think we'll see those common features.
Okay, many thanks, Richard. I think we should stop here and go for the next talk.
Merci beaucoup. Il est temps donc d'accueillir avec plaisir un chercheur français, Thomas Merchet. Bonjour. Donc, tu es chercheur à l'Institut Gustave Roussy et tu vas nous expliquer le rôle de l'ontogénie et des altérations transcriptionnelles dans les leucémies aiguës myéloïdes pédiatriques.
Merci pour l'invitation. Bonjour à tout le monde. Indeed, I will talk about hematopoietic transformation. In the hematopoietic system, there is a need to produce a lot of mature cells of a quite large variety, and it is every day, and it is believed that this is done by a relatively rare population of so-called hematopoietic stem cells that can self-renew the entire life of the individual, but also differentiate into progenitors, and then mature cells to provide these mature cells during the entire time of the lifetime. So this is a hierarchical system, and it is believed, and there are several observations now showing that this hierarchy evolves during development, throughout fetal development, because hematopoiesis is happening in several places in the embryo and fetus. And then it evolves during aging, as it was also alluded to before. just before. For example, a few differences that can be cited are the fact that hematopoiesis is more proliferative in the fetus and is more biased toward giving rise to erythroid and megakaryocyte cells that provide platelets at the end of the day. While in adults, the hematopoiesis is more quiescent, less proliferative, and more myeloid biased. And it is believed that this is to adjust to the need during development, for example, for oxygen transport, and then for developing immune system in the adult. The control of this hematopoietic system is happening through interaction and intertwined mechanism involving extracellular signals and intrinsic factors like the transcription factors that binds to DNA, and their activity is regulated also by chromatin regulators. And it is believed now that these transcription factors organize in multi-protein complexes and that the differentiation is controlled by the different activity of the transcription factors within these complexes. For example, during megakaryocyte differentiation that produce platelets, it is believed that there is a need for replacement of the transcription factor GATA2 for GATA1 that allows expression of genes that are important for terminal differentiation. And this is associated, for example, with expression of the markers, the surface marker CD41 that we will see later. So the importance of this transcription factor in leukemia, in blood cancer, is shown by the fact that many of them are mutated in both myeloid or lymphoid disease, and some of them show a frequency that changes with age. Some of them are more prevalent in young children, some of them later in life. So the question is, why is there this association with age for some of these tumors and for some of these oncogenes? The work of the lab is focused on acute megakardioblastic leukemia that shows a high prevalence in childhood and that shows also a poor prognosis. What we have shown over the years, we and other labs, is that the genetic of these tumors show involvement of fusion oncogenes between transcription regulator. Here, two examples between OTT and MAL, or between ETO2 and GLIS2 due to an inversion of chromosome 16. And these genetic analyses have allowed to associate these tumors to prognosis. And for example, the ETO2 GLIS2 fusion that is quite prevalent is also associated with a very poor prognosis even today. Some of these alterations are associated with other mutations, but for example, for the ETO2 GLIS2 fusion, it's very rarely associated with other fusion, with other mutation. So it appears that it could be the sole mutation in some cases at least. So to understand the contribution of this genetic alteration to disease development, we teamed up with clinical collaborators of the Connect AML network to obtain samples from patients. And we saw that patients with ETO2 GLIS2 fusion show two types of disease. One that is acute megakardioblastic leukemia with a megakardioblastic phenotype and associated with a very young age, and acute myeloid leukemia in other subtypes that generally shows an older age at diagnosis. And to understand the basis of this, we teamed up also with a team of Jörg Schwaller in Switzerland to develop a model in which we can express the ETO2 GLIS2 fusion in an inductible manner with doxycycline. So we can turn on the oncogene and then we can turn it off if needed. What you can see here, if we induce expression of this oncogene in mice, they develop a disease with a large spectrum, some of them being megakardioblastic, some others being myeloblastic, and some are quite immature. So it reproduces a spectrum of what we see in patients. And the question is, why is there this diversity of phenotypes? So what we did was to try to understand, if we express it at different stages of development, what is happening. So what we did was to express it at the fetal liver stage of hematopoiesis, newborn stage or adult stage, and see the phenotype of these cells that we obtain after expression of the fusion that is tracked by two surface markers, CD41 that I mentioned for the megakaryocyte lineage, and GR1 for the differentiation of the myeloid lineage. What you see is that in fetal liver, you obtain exclusively a megakaryoblast phenotype, while in adult, you get progressively a myeloid phenotype. And it is heterogeneous in adult. You get both myeloid and megakaryoblast. So there is also something that explains this heterogeneity in adult. And to understand this, what we did was to take adult cells and purify the different populations that I mentioned briefly at the beginning, the very early long-term hematopoietic stem cells, but also different subtype of progenitors called MPP for multipotent progenitors, and then express the fusion to look at what happens. So what you can see here is that if you express the fusion in HSCs, you get a purely megakaryoblast phenotype, but then if you express it in MPP2, 3, and 4, you progressively get a myeloid phenotype. So what you see here is that depending on the cell you express the oncogene in, you get a different phenotype, either myeloid or megakaryoblast. And of course, the question here is also, does it impact disease development? So we did exactly the same, purify these progenitors from fetal or adult and inject these into mice to see disease development. And what you see here is that from fetal development, here in red, you get a very fast disease development within a few weeks, while when you inject adult progenitors, it takes a much longer latency to develop leukemia. So what you see combined is that depending on the cell type you express the oncogene in, you get different type of phenotype and different aggressivity of disease development. At the molecular level, what we see is that it's associated with different expression of transcription factors. For example, GATA1 is way more expressed in fetal cells compared to adult cells. And it is a mirror effect for CBP alpha that is more expressed in adult. And at the top, you see the mouse model, and at the bottom, you see what the level of expression is in patient, and you see the same trend. And we know that it is functional because when we take cells that should become megakaryoblast, the ones that I mentioned, hematopoietic stem cell expressing ETO2 GLIS2, and then you re-express the CBP alpha, you see that there is a change toward myeloid development. So indeed, CBP alpha controls the phenotype in this setting. So we are trying to further understand the molecular basis of the transformation by this oncogene. What we have done is to understand what are the regions on the DNA that binds this fusion. And we have identified some regions that are called enhancers that control gene expression, for example, the expression of the ERG gene factor. And what we do in an yet unpublished but almost finished work in collaboration with Camille Aubry and Julie Chaumet is to understand how the fusion can bind a particular locus, here the KIT locus, a receptor at the surface of the cell, to control a functional node that allows survival and proliferation of the cell. So together, what we believe is happening at the moment is that this fusion oncogenes binds DNA at certain regions, and that it depends on the cell context, depending whether it's early hematopoietic cell or a more committed progenitors, the region will not be the same. It controls expression of some genes like ERG or KIT, and this is combined allow a multiple alteration of biological processes that contribute to disease development and blockage of differentiation. What I can briefly tell you about what we can think of in terms of targeting these leukemia is as follows. What we observed is that there is a common domain within the ETO family member of co-regulators that is important for tetramerization, for interaction of this protein together. And if you express a small peptide called NC128, you can disrupt this complex. And what we have done was to express this peptide in a PDX model. And we see that when we express it, we abrogate disease development in models. What we need to do now is to improve our understanding of the transformation in human cells. What I have shown you was primarily in murine cells, and we need to do so because these transcription factors bind DNA at specific region, and that the organization of the genome that Olivier Delattre mentioned before is important and different from human to mouse. So I will briefly mention the ongoing work with Erika Brunet and with William Venschenker to develop induced pluripotent stem cell model in which we express the fusion in a very faithful manner because we reproduce using CRISPR-Cas9 the exact genetic alteration. And we know that here you have a proof of principle that we can recreate the mutation in these cells. And when you differentiate these cells into hematopoietic cells, we can reproduce alterations that we see in patients. And we have ongoing in vivo work. And then by doing molecular analysis, here single cell analysis to understand how these cells can differentiate into different type of cells, here erythroid cells, myeloid or megakaryocyte. You see on the left normal differentiation, and on the right you see an abnormal population that we are characterizing now. So I will just leave that for you to read, and I would like to thank all the persons involved in this work. And the last part is done in collaboration also with Françoise Flumiot in CEA and within a consortium that just started, the PEDIAC consortium. Thank you very much.
Okay, thank you, Thomas, for this great talk. So once again, I really encourage you to ask questions on the chat. If you can leave your name and institution with your question, it will be great. So please go on the chat and ask questions. It will be much more dynamic. So I will start the discussion first, or do you have? Okay, I will start first with one question. So you use, Thomas, you introduce your fusion ETO2 GLIS2 into fetal liver and bone marrow. Do you think the environment could be important? I mean, it's very, very different environment, liver versus bone marrow. Is it participating to anything in this context?
Yes, well, as you've seen at the very beginning, I really think that this interaction between environment and transcription control is important, and for the moment we have focused on obtaining these models and characterization of what happens within the tumor cells. But as you suggest, the role of the environment, what type of signal is important to favor disease development is clearly something that we need now to understand better. That's clearly one of the next steps, is to understand how these cells communicate with their environment, and if there is an environment that is more prone or less prone to disease development.
Okay, but did you try to regraft your cells into different environment? Or?
So at the moment, what you've seen here in the presentation was to take fetal liver cells and inject into adult animals.
Only in adults?
They do, yes, because of the system. In fact, in this system, if you induce directly the oncogene expression at the fetal liver stage, the embryo die very fast. So it's not a system that you can use to study this. So you need to graft. What we are going to do now, indeed, is to take these cells and through collaboration with other teams, use micro-injection techniques to inject either at early embryonic stage, newborn, and adult to see precisely what they do in their environment and what the environment is, how the environment is different.
Okay, and regarding hematopoiesis, so you observe in adult that if you introduce into HSC versus MMP, you have different phenotype. Are the MMP different in the fetal liver, very different, beside the level of transcription factor you mentioned? That should explain. Do you see the same trend, perhaps less marked, but the same trend in fetal liver or not at all?
We tend to see a different trend because what I've not detailed in the presentation here, but that was on the slide, was that if you take MPPs from fetal liver, you can get disease development, but you cannot get disease development from adults. So in fact, it indicates that potentially the multipotent progenitors have a slightly different properties in fetal compared to adult. And the hierarchy of the system is probably shaped differently, meaning that maybe the multipotent progenitors are closer to a stem cell in the fetal liver or closer to something that can give rise to erythroid and megakaryocyte lineage, which is here, in this case, the target of this oncogene. So it is possible that this architecture change during ontogeny is responsible for the more prevalent disease in childhood than adult, because then there is less targets, there is less cell targets in adult, potentially.
Okay. Didier?
Thank you very much, Thomas, for this very nice talk. I had actually a question in your fusion-driven leukemia. To which extent you think that the genetic background is an important factor in the transformation of your models? Can you maybe comment on this, or do you have any idea?
I can comment. The short answer would be I have no experimental clue to tell you whether it is important. What we see here is that with a murine model that has a very homogeneous genetic background, we can get disease development. It is possible to use other genetic backgrounds, even for murine models. But clearly, your question is appropriate because very likely the activity of the different, the expression level of the different transcription factors that I mentioned may be different from one individual to another, maybe slightly different, and that could change disease development. And that happens very likely at the transcription factor level, but also at what Celio mentioned is the signals that come from outside, and all this combined is very likely important. So from one individual to another, you may have disease development or not. It's very possible.
Thank you very much.
Perhaps one last question. So you mentioned that the level of CEBP versus GATA1 is very important. Do they collaborate with the fusion in terms of gene expression? Do you see like co-occurrence of their binding site within the target genes of the?
Yes, in fact, we know that the GATA factors are within the complex that binds to DNA together with this fusion, this particular fusion. What we don't know is the dynamic interaction, whether different loci, different genes are controlled the same way. But yes, there is an interaction within the same complex and area of DNA binding, yes.
Okay, thank you, Thomas. I think we should go through the next talk. Many thanks. So now we are going to hear from Charles Mullighan, who is Deputy Director of the St. Jude Children's Research Comprehensive Cancer Center in Memphis. So his work mainly focused on genomic and epigenomic alteration to identify genomic and epigenomic alteration in acute lymphoblastic leukemia and all related diseases. And he's going to talk today about the lineage ambiguous leukemia. So, Charles?
Hello, thank you very much. So the topic of my talk today is to describe some recent work, how we've continued to use large-scale genomic analysis to deconvolute the origins of acute lymphoblastic leukemia, and a recent story focused on the understanding of lineage ambiguous leukemia, one of the least well understood and clinically problematic forms of leukemia. So as is well known, acute lymphoblastic leukemia is the commonest childhood tumor. It's a great success story. Most children are cured, but when relapse occurs, it's difficult to cure. And for that reason, second now to brain tumors, it remains the leading cause of childhood cancer death. And it's also true to say that in the last decade, we've seen a revolution in our understanding of the genomic drivers and classifying events that define different subtypes of acute lymphoblastic leukemia. And it's really RNA sequencing, transcriptomic analysis that's led many of these discoveries. And a couple of important points are that there have been multiple studies, often focused on relatively small numbers of patients, of cases, often up to several hundred, that have identified several new drivers that define different subtypes of leukemia, often involving transcription factor alterations that you've heard some about already in the previous couple of talks, that define new subgroups of ALL. What has been lacking, though, however, has been a large-scale analysis that covers several additional facets, and that is looking right across the age spectrum of leukemia from infancy right through to adulthood, to understand how drivers vary with age, to incorporate whole genome sequencing data, and to look beyond acute lymphoblastic leukemia as well. Now, several groups have performed these analyses, and so this is an ongoing study from my group at St. Jude with a number of collaborators, starting with transcriptome sequencing, and really seeking to use this approach as extensively as we can to classify the disease, and to also to implement it clinically. To summarize a lot of data from this study, this has been extremely powerful, and it's allowed us to move from only appreciating a small number of subtypes driven by aneuploidy and recurrent fusions, where a single fusion defines one subtype, to now in BALL, and the same holds true for TALL, which I'll show some data for later, but in BALL we now appreciate there are over 20 subtypes of the disease, often with more complexity than we previously appreciated. So, for example, many of the subtypes that we now recognize were not seen before because many of the altered genes were cryptic and not seen on cytogenetic analysis. We can see that many of the subtypes are polygenic, where multiple alterations define a group. But we also see that sequence mutations, for example, in transcription factors like PAX5 and Icaros, which specify lymphoid development, can also define specific subtypes of leukemia, and increasingly this notion of phenocopies, where we can see alternate fusions or other genomic lesions that result in a similar transcriptional profile, the standout example there being PH-like leukemia. The limitation of these analyses, as I mentioned, is that many of them are starting from a known phenotype, for example, the B-cell phenotype BALL or the T-cell phenotype TALL, and they don't directly address this problem of lineage ambiguous leukemias that are recognized as being very important. I also make the point that these subtypes we know are increasingly appreciated to be important in terms of outcome. Firstly, the data on the left show that these subtypes vary substantially by age, with a change in good risk and poor risk alterations, with poor risk alterations predominating in older individuals. And irrespective of the age group, these subtypes are strongly associated with outcome, both in children and adults. And so we have a more nuanced understanding of genomic driver and prognosis, irrespective of age. Now, the problem of lineage ambiguous leukemia has been recognized for a long time, that for decades, in fact, by immunophenotyping, that not all leukemias behave neatly according to age. Most leukemias can be initially classified as myeloid leukemia or lymphoid leukemia, and then are subjected to downstream analysis to further stratify the case. But that's not always the case. There are a group of leukemias that are typically classified by non-genomic features, usually immunophenotyping or even cytochemical appearances, and have been classified into different entities such as early T-cell precursor leukemia, acute leukemias of ambiguous lineage, or mixed phenotype acute leukemia. These classifications have been used by default, but they're unsatisfying because they really don't consider underlying biology or genomics. And often the classification schema that are used are arbitrary, they've changed over time, and they're often very similar, and often a single marker can distinguish between two different entities. For example, myeloperoxidase, the myeloid marker, can distinguish between T-myeloid mixed phenotype leukemia and ETP ALL, which are otherwise the same by immunophenotype. And this kind of distinction really makes no sense. And even in further support of this, we now know, as we do understand some of the genetics, that this classification can change within a patient over time. They can be treated with conventional therapy or immune-based therapies, and they can shift between AML and lineage ambiguous leukemia or vice versa, suggesting that this classification is not appropriate. So this has prompted a lot of debate about the cell of origin of these leukemias, and several hypotheses have been proposed, often without a great deal of data. And these are shown here in this cartoon, where there can be leukemias that arise very early that then drive a lineage ambiguous state, so lesions arising in a hematopoietic stem or progenitor cell. Or there can be mutations arising somewhat further down differentiation, that is, in a progenitor that has both T and myeloid potential, or perhaps that the mutation arises in a committed progenitor and promotes dedifferentiation back into a stem cell-like state. All very plausible hypotheses, but often not with a great deal of data to support them. And what I want to show you today are a couple of stories that really, I think, suggest that the leftward proposal that there are mutations arising in an early stem cell progenitor state hold true in these lineage ambiguous leukemias. So the story, at least from my perspective, in terms of understanding the intersection of genomics and cell of origin, began about a decade ago, where colleagues at St. Jude, Dario Campana and Elaine Custon-Smith, at least at St. Jude at the time, they're now in Singapore, described this entity of early T-cell precursor leukemia that was, in some respects, a T-cell leukemia. It expressed cytoplasmic CD3, the T-cell marker, but otherwise had a very distinctive and unusual immunophenotype with a lack of expression of other T-cell markers and aberrant expression of myeloid and stem cell markers. And again, coming back to that point that these cases are negative for myeloperoxidase. If they were positive, they'd be termed mixed phenotype acute leukemia. Now, despite that, there were some similarities noted to the earliest stage of mouse thymic T-cell development, the double negative 1 stage, or the early T-cell precursor, and hence the terminology that this was called an ETP ALL. But several lines of evidence suggest that this is not a T-cell leukemia at all. Firstly, if one looks on the left here at the mutational spectrum, one sees a very different pattern of mutation than one sees in typical T-cell leukemia, with lack of mutations of typical T-cell genes like Notch and CDKN2A, for example, but positivity for a suite of mutations that are much more myeloid leukemia typical, mutations in myeloid hematopoietic transcription factors, cytokine signaling pathways, and chromosome modification, particularly PRC2, that again are more common in myeloid leukemias. And then if we did an observational analysis comparing the transcriptional profile data to that of a normal human ETP, normal human true early T-cell precursor, we actually find that there is negative enrichment, that the leukemias do not look like a normal ETP at all. In contrast, they look more like a normal hematopoietic stem cell, or indeed a malignant myeloid hematopoietic stem cell from a variety of different analyses, suggesting that at least on this observational data, the cell is locked into an HSC type state. But the question remains, how did it get there? Did it start as an HSC, or did it perhaps back differentiate from a more committed precursor? So there are two more recent stories that shed some light onto this more definitively. And the first is a published story that, again, comes back to some of the data I was showing you before from the BALL analysis, where one of the new subgroups that we and others identified with this group shown here with rearrangements of a gene called ZNF384, a zinc finger transcription factor that has a role in normal bone development and other lineages, but not the lymphoid compartment, that was defining a transcriptionally very distinct group of leukemias with a number of fusion partners involving epigenetic modifiers and transcription factors fused with this gene ZNF384. Now, in parallel, we had been conducting a study with the goal of better identifying the biology of mixed phenotype acute leukemias, again, starting from a somewhat restrictive position where we were starting with a cohort of cases that were defined as MPAL based on their immunophenotype, and then taking these into downstream genomic analyses. And there were several lessons learned from this study, but the one that's relevant here is that if we look at the cases that are B myeloid, the cases in the red box there that are defined by the B cell marker CD19 and a myeloid marker, typically myeloperoxidase, over half of those cases express the same fusions as I showed in the previous study of BALL, fusions of ZNF384 with these transcriptional regulators. If we look further at these cases and we compare all the ZNF384 rearranged cases, whether they're diagnosed as BALL or mixed phenotype leukemia, their concomitant genetic alterations are the same. And if we look at their transcriptional profiling here, another Tisney plot showing gene expression data, these cases are homogeneous, they have the same gene expression profile. So from all intents and purposes, and now actually confirmed with mouse modeling that we've done that I won't show today, we know that these leukemias are the same, whether they're diagnosed initially as BALL or mixed phenotype acute leukemia. So why is there this phenotypic plasticity, if you like, where either one has a single population of cells that expresses B and myeloid markers, or as is often the case we see within a patient, they can express multiple subpopulations by flow cytometry. Two examples are shown here for a B myeloid and a T myeloid case. It's not genetic, and we address this by purifying from many cases these immunophenotypic subpopulations and sequencing them, and found that the concomitant genetic alterations were the same in each subpopulation. So this variegation within a case is not driven by genomics. In contrast, if we do a similar experiment, that is, if we purify the subpopulations by flow and we then transplant them into immunodeficient mice, we see that these subpopulations can then reconstitute the immunophenotypic diversity of the primary tumor. So that was a very important observation that indicated that this lineage ambiguity and this lineage plasticity is inherent to every tumor cell. It's not because of some terminal differentiation event. And again, I come back to the point I made before about what we see in patients, where often these patients are treated with leukemia, and again, they can shift between mixed phenotype leukemia and ALL or AML and vice versa. So this plasticity is inherent to the tumor cell. So now some insights that these leukemias appear to be a seed and soil where we have the right genomic alteration arising in a hematopoietic progenitor cell. This is clinically relevant. If we look here, for example, at the gene expression profiling of these leukemias, here we see that one of the top deregulated genes in these leukemias is FLT3 from non-mutational mechanisms. In fact, if we look at the levels of FLT3 right across the landscape of ALL, we find these leukemias have the highest level of expression of any leukemia. And indeed, patients have already taught us lessons as to the targetability of this finding, where there was a notable case published by Tim Lay and the WashU group of a patient that had one of these fusions of ZNF384 fused to P300 and was noted to have very high level of expression of FLT3 through non-mutational mechanisms. And then when treated with sunitinib, there was a dramatic response and the leukemia was induced into remission. So a nice example where we can see a precision medicine vulnerability, if you like, but not through a true mutational fusion targeted approach, but through an alternative signaling pathway targeting approach. So these findings were starting to shed light on the biology and drivers and cell of origin of these lineage ambiguous leukemias. But they had two main constraints. These studies were firstly starting from cohorts of patients that were initially defined by immunophenotype, B leukemia, T leukemia, or indeed mixed phenotype or ETP. And they were predominantly based on RNA sequencing-based data, which, while very informative, does not have the power to identify all genomic alterations that may be relevant. Of course, RNA sequencing has only limited potential to identify all structural alterations and non-coding alterations. So with those findings in mind, we took the step of performing a pan-leukemia analysis where we collected as many leukemia samples as we could that had suitable RNA sequencing data and often whole genome sequencing data to try and fully understand the spectrum of subtypes of leukemia. And as I'll show you today, in part of this study with a strong focus on defining these problematic lineage ambiguous leukemias. We took a curated set of cases that we'd studied before of BALL, drawing down now from about 3,000 cases to 1,400 representative, AML cases, as many mixed phenotype cases as we could, and a large number of T lineage leukemia cases as well, and subjected these to a large number of downstream analyses that for reasons of time I won't go through today in detail. So this shows an overall t-SNE plot, gene expression-based t-SNE plot, where each case is color-coded by subtype, by genetic subtype, and by lineage. And one can immediately see here the B lineage cases. The actual layout is somewhat different, but the themes are exactly the same as I was showing you before, where the conventional BALL subtypes largely cluster in their own group. And then we have this group up here with the more myeloid leukemias and T-cell leukemias segregating differently. So now to dive deeper into this, we excluded the conventional BALL subtypes and then worked with a residual group of cases. And that's shown here. So this is a t-SNE projection of about the 1,100 cases, a little more than that, of non-B-cell leukemia samples. And we can see multiple of the previously identified subgroups. If you look to the more black subgroups on the left, the myeloid leukemia subtypes, the more green subtypes on the right, the T-cell leukemias. But a number of new subtypes fell out that were not previously identified, and the subtype that I'll focus on today is this one here, where we saw a group of about 62 cases with a very distinct gene expression profile that you can see here. In an integrated analysis of allele-specific expression, all of these cases had monoallelic expression of BCL11B. We subsequently showed, as I'll come to in a moment, that they have structural variations deregulating BCL11B, and the vast majority of these cases have concomitant activating mutations of FLT3, a commonly mutated gene as seen in myeloid leukemia. Importantly, this group, while very genetically distinct, is very similar to what I was showing you before for ZNF384, where it transcends a conventional immunophenotype. So this included about 1/3 of T myeloid mixed phenotype cases, 1/3 of early T-cell precursor cases, and a smattering of AML and undifferentiated leukemias as well. We went on to further confirm this, so we studied a group of T and mixed phenotype leukemias from an adult leukemia cohort and found exactly the same thing, that about 1/3 of T myeloid and ETP alleles belong to this group. And it has what we would call a fairly strict ETP immunophenotype, where with the same kind of ETP features that I showed before. But a key thing here is that MPO is variable, and that accounts for the cases being diagnosed as either ETP ALL or T myeloid leukemia. I'd now like to show some of the genomic data because I think it's instructive into understanding the biology and also the cell of origin. So the first is to look at the actual organization of BCL11B and the positions of the breakpoints. And so here we see the BCL11B locus on chromosome 14q, and each row here shows the position of breakpoints of partner genes, and we've identified at least 8 partners of rearrangement that appear to deregulate BCL11B, and each of the black lines is referring to the position where the gene breaks adjacent to BCL11B. And one can see that all of the rearrangements spare the coding region of BCL11B, with a couple of rare exceptions that are actually fusion chimeras, but basically allowing deregulation of the entire BCL11B locus. So what could this mean? I mean, BCL11B is by no means a stranger to leukemia biology. It's a transcription factor that's normally restricted to the T cell lineage. It's absolutely required to some of the earliest stages of T cell lineage specification and differentiation. And it's known to be a target of genetic alteration in TALL, either through mutations and deletions that affect DNA binding zinc fingers that are presumed to be loss of function, or as an enhancer donor, specifically the rearrangement with TLX3 that describes a specific subtype of conventional TALL. So this suggests that there may be a dichotomy for the role of BCL11B in promoting tumorigenesis. Furthermore, if we look at the level of expression of BCL11B in normal cells, it's expressed at high level in normal T cells as shown here, but it's absent in hematopoietic stem and progenitor cells. The presumed cell of origin of these lineage ambiguous leukemias, suggesting that if BCL11B is deregulated, this could be through some aberrant hijacking or enhancer activation event. So to analyze this, we performed integrated transcriptomic and epigenomic analysis, including K27 acetylation analysis, high chip analysis for K27 and topology, BCL11B chip-seq, and a number of other marks in various cell types, so normal T cells, CD34 cells, T cell leukemias, as well as these BCL11B rearranged lineage ambiguous leukemias. And the first point to show here is that if we look at data of K27 acetylation from normal CD34 hematopoietic stem and progenitor cells, we can see that there is a peak of K27 acetylation right in the hotspot of rearrangements of one of the partner genes, one of the commonest ones, a rearrangement cluster downstream of the ARID1B gene, as shown here. This is a super enhancer, and in fact, if we do a formal super enhancer analysis of CD34 cells, we see that all of the positions of rearrangement are super enhancers in normal CD34 cells. So this was a very striking observation, suggesting perhaps that these super enhancers that are normally active in the earliest hematopoietic stem and progenitor cells are being hijacked and rearranged to BCL11B. Now that's just one example. We saw the same thing with all of the other rearrangement partners, another one of which is shown here. This is the CCDC26 or B-cell enhancer or BENC enhancer that's located downstream of MYC on chromosome 8. This is a very important super enhancer region in hematopoietic development as well as malignancy. Normally it's rearranged and activates MYC, but here it's rearranged to BCL11B. So how can we integrate the chromatin marking as well as chromatin organization analysis? And we use this using the high chip approach, which couples chip sequencing for a given mark, here K27 with chromatin topology. And so again, these are normal CD34 cells here, showing no evidence of K27 acetylation or RNA sequencing at BCL11B in normal stem cells. But in contrast, we see this area of activity of acetylation and expression adjacent to one of the partners that I showed you before, ARID1B, but no interaction between the two. In contrast, if we look at this case with a rearrangement, we see both high-level acetylation and RNA sequencing activity at the BCL11B locus and very clear evidence of looping between this hotspot of rearrangement adjacent to ARID1B and BCL11B itself. We saw this repeatedly with every primary sample that we studied, confirming this mechanism of rearrangement between a stem cell enhancer and BCL11B. But in addition, we found that a substantial proportion of these cases, 20%, had a different mechanism with generation of a novel enhancer downstream of BCL11B. So again, this shows the BCL11B locus and about 700 kilobases downstream, a very small region, 2 kilobases, that undergoes high-level tandem amplification to about 15 to 25 copies as shown here, with a comparison case at the bottom there showing no evidence of amplification. We confirmed this by genomic quantitative PCR, showing this very stable level of amplification. So there appears to be a sweet spot of the number of copies of this gene that's required to facilitate enhancer activation of BCL11B. And indeed, this is accompanied by some evidence of gene expression here. And in fact, this is a biomarker for the presence of this amplification with some RNA amplification signal, expression signal seen in this region of amplification. We were interested to understand the mechanism of this amplification. Is it an episomal event that might be inserted elsewhere in the genome or acting as a true episome? And we turned to PacBio long-read sequencing in 2 of these cases. And firstly, we were able to show that at each end of this cassette, there are repetitive self-chain sequences that are a sequence that's used for germline amplification in normal development. But also we could show with long reads that spanned the entire region of amplification in these cases that this is a true tandem array of these cassettes, one after the other, up to 20 to 25 copies that is occurring in situ downstream of BCL11B. So this then generates a strong enhancer. There's a tiny little amount of K27 acetylation in normal stem cells, but in the context of this amplification event, we see very strong K27 acetylation and strong activation of BCL11B. And indeed, if we compare this to other cases as shown here, there are 2 cases with this amplification on the left showing this extremely high level of K27 acetylation compared to essentially nothing in the comparison cases shown on the right. If we then turn to the high chip that I was showing you before, the results here are very striking, where we see in the center, we see this region of amplification showing very strong looping and activation of the entire BCL11B locus. And indeed, there's also looping to the right because it's co-opting a second T cell enhancer, the ThymoD enhancer, into this combined transcriptional activating network that drives aberrant BCL11B expression. Now, coming back to human data again, just to complete the talk, the question remains, how do the leukemias compare to normal stem and progenitor cells? And so we collaborated with John Dick, who had developed gene expression and ATAC-seq maps of normal human hematopoietic stem and lineage development. And the data shown here on the top of the slide are taking our entire pan-ALL or pan-leukemia cohort and looking for enrichment of specific signatures. And we can see in the middle at the top, the HSC signature is enriched just in the top few cases there, and they're marked in green on the right. So the BCL11B group are expressing a normal human HSC gene expression signature. And if we then go on, as we did, and we performed single cell sequencing with combined gene expression and ATAC sequencing, and compared this to the ATAC sequencing profile of normal hematopoietic stem and progenitor cells, we see the converse that makes sense. We see very strong enrichment of a normal HSC signature of chromatin accessibility and gene expression in these BCL11B rearranged leukemias. So now we have very strong evidence that not only do these leukemias arise from BCL11B hijacking, they share features of hematopoietic stem cell, but they also have evidence from integrated genomic and epigenomic analysis that they are arising from these events occurring in an immature hematopoietic stem and progenitor cell. And finally, the experimental proof is to express these lesions in hematopoietic stem and progenitors. And we see that both initially in transducing mouse lineage negative cells, we see co-transformation introducing BCL11B in the co-lesion FLT3. And at the bottom in the red box here, when we transduce normal cord blood CD34 cells, we see the induction of lineage aberrancy and emergence of lineage ambiguous populations. So these are my conclusions, that gene expression profiling identifies this new subtype of leukemia defined by BCL11B deregulation. They cause juxtaposition of BCL11B with hematopoietic super-enhancers. Chromatin topology results that analysis shows that these SVs result in enhancer hijacking. And this is yet another example where genomic alterations truly transcend previous canonical subtypes of leukemia and should be used to understand disease. The founding lesions arise in and co-opt and indeed corrupt a hematopoietic stem cell program. the final point I would make here is that there are therapeutic options for lineage ambiguous leukemia. I won't go into these today, but in fact, there are at least 2 that we have evidence for already, that FLT3 inhibitors can be active. And indeed, the new BH3 mimetics showing such promise in myeloid leukemias are also highly active in these very early BCL11B lineage aberrant leukemias. And so there I'll stop. Much of the work I've shown you today was driven by Lindsay Montefiore and Xiaohui Gu in my lab, as well as many collaborators, both at St. Jude, and key collaborators around the world, particularly the Munich Leukemia Laboratory and Claudia Hafelach and John Dick in Toronto. So thank you for your attention.
Okay, thank you, Charles. So we have a question from the chat. I will try to read it. So you have shown in your single-cell RNA-seq data that B-ALL leukemia are very heterogeneous, but AML and TLL cluster kind of together. Do you think that finally during hematopoiesis and T and myeloid lineage are more related than T and B lineages, questioning common lymphoid progenital cell existence that can also explain why ETP-ALL can harbor myeloid future? Did you get the question? Or?
I think so, thank you. Yes, thank you for the question. Just to be clear, the initial TISNE plots I was showing were not actually single cell data. They were bulk transcriptional data where each case was shown as one point. So it kind of looks like single cell data, but actually it was bulk. But anyway, that notwithstanding, one has to be a little careful because I think once one includes cases from all lineages, the overall lineage can cause compression of heterogeneity, and that's the reason why we actually excluded the conventional BALL groups before going into the downstream analysis. As I showed, once you take out the BALL cases, one sees much greater separation between the subtypes within the T and myeloid group. However, again, your question about the cell of origin and progenitor. So yes, I think that the cell of origin doesn't follow the strict hematopoietic hierarchy that's been acknowledged for a long time, at least that described in mouse where there's early divergence between a myeloid and a lymphoid progenitor. I think the human data are now strongly supporting what's been proposed for human development from John Dick and others for a long time, that there is retention of multi-lineage potentiality until relatively late in development. And there are multiple progenitors that retain both T and myeloid or B and myeloid potential. And if one has transformation of those, one can then generate at least some of the lineage ambiguous leukemias. Although, as I've shown you, some of them arise from an early stem and progenitor cell. You specifically mentioned ETP-ALL, and even that is heterogeneous. You know, only 1/3 fall into that BCL11B group, where we strongly believe the cell of origin is an early hematopoietic stem and progenitor cell. But there are other ETPs that are much more like a typical T-ALL or much more like a myeloid leukemia. And for those, the cell of origin may be quite distinct, but I don't have direct evidence to support that at the moment.
Okay, so regarding single-cell RNA-seq, if you had performed single-cell RNA-seq, do you think that the mixed tumor could have like different population? One will be closer to T-ALL or B-ALL and the other to AML cells? Did you?
Yeah, that's a great question. And in fact, for the cases that we've studied, that's not what we've seen. You know, we've used a variety of different single-cell profiling approaches to look at expression, to look at ATAC, and also to do, you know, targeted interrogation of fusions and co-mutations. And that's a bit more challenging in terms of sensitivity. But anyway, if we look at the single-cell data for this group, the BCL11B cases, it's actually quite interesting because one sees very homogeneous expression of BCL11B. So it's aberrantly expressed, but it's in pretty much all cells. But MPO expression is really variable. It's only on in some cases, it's on in all cells. In some cases, it's only on in a subset. And I think that doesn't suggest that you've got different populations of cells, because apart from that marker, we can't pull out different populations that are more T or more myeloid. It just suggests that it's a really variable marker. Now, there may, of course, be cases in single cell where we do see much clearer variability within the cases, but we've not seen that yet. But you know, this is an ongoing project, so it will be interesting to look at some of the other cases as time goes on.
Okay, another question from the chat. Please, when you ask question from the chat, if you can like identify yourselves, that we can like mention your name and your institution, it will be great. But continue to ask question on the chat. So do you see? So this question is the following: do you think that like for AML, for TILL as well as for BALL, HSC is also at the origin of the disease, explaining why you have enrichment of stem cell signature in your data set?
Well, yes. I mean, for this subtype of leukemia, an early HSC really appears to be the cell of origin. But I don't think that's the case for all leukemias, and I don't think it's the case even within the lineage. So there are, for example, if one looks at B-cell leukemia, one does have a surrogate footprint where one can look at antigen receptor rearrangements, for example. And we've also done backtracking from patient samples where one can take out the normal hematopoietic stem and progenitor populations and do targeted mutation or fusion detection analysis. And one can see there is heterogeneity in the cell of origin. Some appear to be later and some appear to be earlier. That, at least in our hands, that work is ongoing for myeloid leukemia. We are now doing those analyses for AML, but experimentally we have evidence they're also arising in different cells of origin also. In TILL, there's heterogeneity. There are nice data from Jan Kuhls in Leuven that suggests that in some TILLs, you can detect the founding mutations in an early progenitor, at least a CD34 positive cell. In others, you cannot. So I think it's a little reductionist just to say that in all of these, an HSC is the cell of origin. I think it does vary, and that's why we see heterogeneity in the disease.
Okay, one last quick question, Thomas.
Yeah, Charles, your BCL11B study is extremely nice. I may have missed it, but do you know what is below this amplified region in terms of DNA binding pattern? Is it the typical so-called heptad complex, or do you see something else?
Yeah, Thomas, thank you. That's a great question. So we have some data, we have some evidence for some CBP, RUNX, and other factors that are under that region, but it's a little incomplete. We need to do more work, and we're coupling both the kind of chromatin occupancy analysis with, protein complex purification to see what's in the complex with BCL11B, and comparing that between conventional TILL and these leukemias to see whether it's just co-opting of the same network of BCL11B or whether the complex for switch-NIF or other reasons, for example, may be quite distinct. So we have some insights, but a little immature at the moment.
Okay, I think we should stop here. Many thanks, Charles, for this great talk.
You're welcome. Thank you.
And I think we should move to the next talk.
Yes. Je vous présente donc la dernière présentation. Elle est faite par Didier SURDEZ. Tu as longtemps travaillé dans l'équipe d'Olivier Delattre à l'Institut Curie. Tu inities dorénavant une nouvelle équipe à Zurich et tu vas nous présenter le rôle de l'altération STAC2 dans le sarcome d'Ewing.
Tout à fait, merci beaucoup. So, which is about deciphering the function of STAC2 in Ewing Sarcoma. So, Ewing Sarcoma is the second most frequent pediatric bone tumor, so it peaks around the age of 15, and it is characterized by the characteristic translocation involving FAT-ETS gene member, but the most frequent is the fusion between EWS and FLY1, but was described back in 1992 by Olivier, and you may have seen his introduction on this sarcoma, and this leads to the expression of a chimeric oncogene deregulating the transcription of the cell of origin. Now, being a pediatric cancer, there are few recurrent alterations, but we and other groups have managed actually to identify some recurrent alteration in Ewing sarcoma. So CDKN2A was identified, TP53, but the most recurrent was STAC2. So what is STAC2? STAC2 is part of the coenzyme. The coenzyme is a ring, which is actually very important, for instance, to shape the 3D structure to generate this loop structure of the chromatin DNA. And within this loop, you can actually have interaction between promoter here in blue and enhancer in red region. And importantly, I should mention that STAC2 bridges the different key members, which has SMC1, SMC3, and RAD21 of this coenzyme ring with a CTCF, which is known to generate this kind of loops. So STAC2 is not only mutated in Ewing sarcoma, but is also mutated in some other more frequent adult tumors, such as bladder cancer and glioblastoma. To decipher actually the function of this loss of function mutation, we use isogenic model, and we started with Ewing cell line, which are proficient for STAC2 expression, and using CRISPR-Cas9 technology, we generate the STAC2 knockout. We also managed to rescue these knockouts. And I should here mention that we also generated a STAC1 knockout, which is the paralog of STAC2. And we did this in several models. Of course, these are isogenic clones, and you can criticize that these are clonal effect. And we therefore also generated with a panel of other Ewing cell line using siRNA to see bulk short-term effect of the loss of STAC2 in this model. Now, if you perform transcriptomic analysis of in the isogenic model, you will see a core signature of roughly 500 genes which are modulated. And if you now look at this core signature in the different siRNA experiment, you can see, of course, that in the parental A673 and TC71 model, this is identified again, but you can also see that the signature emerged as soon as 24 to 48 hours of inhibition, and that the inhibition, especially for the downregulated genes, is also observed in the different Ewing cell line models. Now, going a step further, we perform gene set enrichment analysis, and we identify among roughly 19,000 signatures 5 Ewing signatures in the top 20. And this was not only true in the siRNA and isogenic models, but we could also see this strong activation signature in a panel of large panel of Ewing cell line and also Ewing sarcoma tumors, which are wild type or mutated for STAC2. So the message here is to say that when you have STAC2, E+fly activity is very high, whereas when you have a mutation, the activity, the transcriptomic activity of E+fly is reduced. But actually, this was a bit contradictory to this experiment that we perform using ChIP-seq, where we look at the binding of E+fly in the wild type and mutated condition and barely see any difference between those two conditions. That was true for E+fly binding, but also for H3K27 acetylation enhancer marks. And you can also appreciate that the CTCF and the different coenzyme members were not really affected by the loss of STAC2 expression. For the sake of this presentation, I will actually use DKK2 as a prototype gene locus. And here you can also appreciate that in black wild type and red STAC2 mutated, the binding pattern at the CTCF and the coenzyme member, as well as the different E+fly enhancer, which are here a microsatellite of GGAA, classical active region in this Ewing sarcoma, but the pattern between wild type and mutated is almost unaffected. And this is, of course, in contradiction of the transcriptomic data, where you look basically here in every single experiment, DKK2 is decreased upon the STAC2 mutation. So how can you actually explain this? And to actually understand this concept, you, of course, have to remind that if you look at ChIP-seq, this is a unidimensional vision of the genome, and which is absolutely not the case in the nucleus, because we know that chromatin and DNA are folded and generate kind of loop structure. So to investigate this, of course, you can perform IC, and IC will allow you to identify all type of interaction in the nucleus. However, since we knew that STAC2 interact directly with the CTCF and coenzyme, we particularly focused on the interaction that were involved in the CTCF coenzyme complex using iChip for CTCF. And here I'm representing a frequency interaction map at the DKK2 locus using Hi-C, where you can see the different interaction in this region. The more red, the more interaction you have. And now if you perform iChip, I hope that you can appreciate that the interaction contact were actually dramatically increased at the site of CTCF. But not only they were increased at the site of CTCF, but you can probably see this kind of stripe pattern that actually were lost upon STAC2 KO knockout, and that we could actually restore upon re-expression of STAC2. And if you now look in the STAC1 knockout, indicating that you only have STAC2 in these cells, at the opposite, you can see that the intensity of this stripe is much higher. So how can you explain this kind of stripe pattern? So it's now generally accepted that this kind of structure occurs through a process, a dynamic process that is called anchored chromatin extrusion. And in this process, basically, one side of the chromatin is anchored to the coenzyme CTCF, whereas the other sides extrude, the chromatin extrude through the coenzyme ring. And if you have one anchor at the left, you get this kind of X stripe. And if you anchor on the other side, you can get this kind of Y stripe pattern. So what I'm telling you now that it's STAC2 appear to reduce the intensity of this stripe. However, we had no tools that allow to identify this kind of strike in the genome, and we implemented a bioinformatic approach that I will not detail here, but you can see that with this black line, we can quite faithfully detect this kind of stripe pattern. And if you now compute the intensity of this stripe in the STAC2 wild type and the STAC2 knockout, you can appreciate here that in the mutated cell line in red and green, the stripe are reduced genome-wide. And this was true also at short term in our siRNA experiment. And as mentioned and illustrated before, if you now knock out STAC1, only have STAC2 expressed in the cells, these stripe patterns are really increased. So far, I've shown you that STAC2 appear really to be important to anchor the coenzyme CTCF and to allow for asymmetric extrusion of the other extremity. And when you lose STAC2, basically, you reduce this anchorage, loosen the both chromatin strands, and actually we wonder if this anchorage would not favor the promoter-enhancer interaction, and if by loosening this anchorage, the promoter and interaction would not be weakened sequentially. So for this, we then perform iChip for a classical enhancer mark, H3K27, and now again, through bioinformatic analysis, we regenerated chains of promoter and enhancer in these cells. And as illustrated at the DKK2 locus, you can nicely see that we were able to identify this promoter and enhancer between at this locus. But we could also go, of course, one step further. And now along these promoter and enhancer chains, we looked at the signal intensity of H3K27 acetylation in our ChIP-seq data. And actually, as illustrated in the beginning, we couldn't really see much difference, meaning that H3K27 acetylation mark is there. However, the capacity of this promoter and enhancer to interact together, and as you can see here in the iChip data, was also reduced by approximately 50%. The next question was, of course, to ask whether this mechanism is a general mechanism, or is this only occurring at E-fly, since E-fly mostly occur at enhancer. And as illustrated here, it appears that the loss of STAC2 is independent of the fact that E-fly is present at this enhancer or not. since we can see a similar loss of interaction in these two kinds of chain of interaction. So here, to go to the next step, there are now quite a lot of literature mentioning that E-fly is a major driver of the migration phenotype. And if you reduce the transcript of the protein level of E-fly in a Ewing sarcoma cell line, this promotes metastasis and migration. So here we don't affect the transcript of the protein level of E-fly, but we affect his cis-mediated activity. So we wonder actually if this would also lead to an increased migration of these cells. And as you can see here in this migration assay, the STAC2 mutated cells would close the wound faster than the parental wild-type cells, as you can quantify here. And we perform this also in short-term siRNA experiment and could reproduce this also in different Ewing sarcoma cell line. And we finally use a Ewing sarcoma model in which STAC2 is mutated, and using the doxycycline-inducible system to express STAC2, we were able to slow down the migration of these cells by expressing STAC2, whereas the empty control was absolutely not affecting the migration speed. Now, to go a step further, actually, what are the mechanisms that are driving this migration? As I told you, E-fly is a key factor in this migration. So we wonder if we now use STAC2 knockout cells and we do overexpress E-fly, would we impair the migratory capacity? And this is what you can see here. This is the wound healing closure speed. So using cell line with an empty vector in the yellow curve, and now overexpressing E-fly nicely restore the original slower migration capacity of these cells. So altogether, what I would like to, what I told you today is that actually STAC2 appear to be a key factor for the anchorage of the coenzyme CTCF ring, and that this anchorage facilitates downstream promoter-enhancer interaction, in which E-fly is key in this Ewing sarcoma. And actually, when you mutated STAC2, what you can see that you have loss of this anchorage, with loosen both chromatin hands and disrupt promoter-enhancer interaction, and also impair the E-fly cis-mediated activity and favor migration of these cells. So with this, I would like to acknowledge key people in this work, and of course, my mentor, Olivier Delattre, and his Interlab. And I would here also like to mention Sandrine Grosstedt, Véronique Hill, and Sakina Zaidi, that have been really key in this work. Also like to thank our collaborator and the group of Erika Brunet, of course the funding agency, and especially INCA here, and of course patient and family, and the different charities. And last but not least, I would like also to thank my current institution, and just mention that some postdoc positions are available in the lab that I just started. And thank you very much, and happy to answer any question.
Okay, thanks, Didier. So I will start a bit with some questions. So yeah, so you mentioned that the frequency of Ewing sarcoma is higher in the Caucasian population. Do we have any clue of what happened?
Yes, so this is a bit of different question, of course. So the background, maybe I should mention that it is known now for several decades that the incidence of Ewing sarcoma in the Caucasian population is higher than, for instance, in the Afro-American population. And the United States, that we know that this is not due to any environmental factors, as the people that live in the same country basically don't show the same incidence. But so what is important here, how it can happen actually. So the genetic background in Ewing sarcoma has been assessed using genome-wide associated studies, and we have shown actually that at least 6 different regions, which are most of them are vicinity in this GGAA microsatellite region. And it has been shown by the paper of Thomas Grunewald that at least this region in the vicinity of the EGR2 is important for the development of Ewing sarcoma. So probably the genetic background and the different polymorphisms that you can have at this GGA microsatellites is key indeed for the development and to allow the binding of E+fly at this initial states.
Okay, I will continue a bit. So according to your model, we may expect no difference in terms of target genes between the STAC2 versus wild type versus mutated. Only the intensity of genes that is decreased, right? Is what you observe exactly or not?
I mean, the transcriptome is really reshuffled, right? We have thousands of genes that are differentially expressed between wild type and mutated in the isogenic model. But really what is key is the fact that this is probably driven by the fact that enhancer and promoter interaction are impaired in the context of STAC2 mutation.
So you have mostly downregulated genes in this?
So I have to say that we have mostly focused on downregulated genes because this was the most obvious signature. But I mean, this...
And all these downregulated genes are target genes of EWS?
Yes. Most of the downregulated indeed are target gene of E+fly. Now, we didn't really focus on the aspect of upregulated genes.
That was my next question.
I mean, this work had just been published officially this week, and we published actually a back-to-back paper in Cancer Cell with the group of Kim Stegmaier, and actually they've focused also a bit on the genes that are upregulated. And there actually something that we also had seen but didn't describe in this paper, that probably the genes that are upregulated, there is a mechanism at the promoter where this gene lose the repressive H3K27 trimethylation mark. So it's not the entire story, but this is the story we mostly focus on.
Yeah, so it's very genome-wide, like a mechanism that you lose either repression or activation, whatever it's dependent or not on EWS fly genes, right? Correct?
Exactly.
Okay. And so my question is so you see only mutation in STAC2, so obviously not in STAC1. What would be a mutation in CTCF or something like that? Do you have any?
So I think it's a very important question. So first, STAC2 is in chromosome X. So and STAC1, I think it's chromosome 6. So if you have the activation of STAC1, for instance, in the girl, of course, this leads to complete alteration of the expression. Now, what still remains a mystery is, for instance, why in some leukemia you see all members of the cohesin member. If you look at AML, for instance, you have 25% of STAC2, 25% of ROD1, SMC1, SMC3. Why is this the case, and why in Ewing this is still a mystery? And yes, so this is something that we still should investigate.
Do you have a question?
Didier, do you think that so you see some kind of flexibility in the genome organization due to STAC mutation? Can we say this, some kind of loosening?
This is still iChip, this is fixed cell, so this is, yes, what we observe is that, of course, on a bulk population, millions of cells. And I think one of the big challenges, and that will be probably the next step, is to be able to go at single loci in nuclei to look what is really the dynamic of this chromatin extrusion. So technically, we are not really there, but this is something that I'm really interested to look indeed how much we can see this anchorage, and we can see both actually chromatin hands moving in and out of this chromatin loop. And maybe I should also mention that here I just show you the DKK2 locus, but what we have seen here in our data that there is really a broad heterogeneity in terms of dynamic of extrusion, meaning sometimes we have very strong stripes, sometimes the beginning of stripe is very strong, and the corner peaks, which means the steady state, is also more intense. So there's really plenty of research to be done in that direction to understand, but that will probably require to go in single cell imaging, single loci. Awesome. Yeah.
But maybe do you think that this flexibility allows to adapt gene expression better to different environment and generate, some kind of plasticity that allow to adapt?
Maybe to go back to the different topic of tomorrow's presentation, I didn't mention, but STAC2 genome-wide screening, looking at what allows for self-renewal of hematopoietic stem cells, identify actually STAC2 as a key members, right? So if you knock out STAC2, you favor the proliferation of hematopoietic stem cells. And of course, we know that the more you go towards stem cells, the more you have the chromatin that is open. So indeed, it would be quite interesting to dig also into that direction.
So there are questions on the chat, but I think we can keep them for the general discussion in French.
Sure.
Okay, so there is one question. I just want to finish with one more question. Also, we are like, so I think it's very counterintuitive what you are telling us. The way that you are telling us, it's like basically we need to lower down the activity of EWS Fly1 to get metastasis. Is it that very surprising?
It of course.
So it means that almost you are like lowering the oncogenic hit, making the cells more close to the cell of origin? Are they very migrating cells, or what is your idea about that?
I was not the first one to actually show this, because I think the pioneer work from Olivier and some like Georges-Alain Franzetti showing that this fluctuation of EWS Fly transcript level was key to induce the migration. And of course, it was difficult to sell, because you want to say that reducing an oncogene may favor the migration, but I think this is now more and more accepted, and I'm pretty sure, I mean, we have shown this in Ewing Sarcoma, but there are now evidence that this is probably also true in many fusion-driven cancer, yes.
Okay, thanks, Didier. So we are going to go to the main discussion. So this aspect is going to be in French. So I will switch in French, but before starting, I mean, we will have a English translation for non-French speaking people. So stay with us, and also during this discussion, we hope to have many questions and to discuss as much as we can this topic. So avant de commencer, on va faire un petit rappel en fait on va essayer d'aller assez vite pour rappeler un peu des notions un peu générales, et on va faire des petits rappels sur les différents talks qu'on a entendus pour vous aider à poser vos questions. Élise,
merci Celio, merci à tous pour ces présentations et ces échanges riches. J'invite effectivement les familles, les parents qui nous écoutent à poser leurs questions à nos chercheurs. Ces temps de parole sont rares et sont importants. Je vous présente plus Celio, qui est chercheur sur la cancérologie pédiatrique à l'Institut Curie et qui va nous expliquer une notion importante pour la compréhension de la genèse des cancers pédiatriques, pour la compréhension de vos travaux, une notion notion qui d'ailleurs a été évoquée à multiples reprises sur les précédents talks, la notion de cellule d'origine. Alors Celio, qu'est-ce que la cellule d'origine?
On va commencer par une petite présentation un tout petit peu générale. Donc l'idée, c'est de faire de vous donner des idées très très simples pour alimenter la discussion. Donc on va venir un petit peu sur la notion de cellule d'origine. Donc déjà, les cellules sont les éléments des différents organes. Donc nos organes sont constitués de différents types de cellules. Par exemple, le cerveau, on sait très bien qu'il va y avoir des neurones. Dans le sang, on va avoir des cellules immunitaires comme les globules blancs, les globules rouges. Donc tous nos organes sont constitués de cellules et ces cellules sont différentes même au sein d'un même organe. C'est-à-dire qu'on ne va pas avoir que des neurones dans le cerveau, etc. etc. Donc un petit rappel rapide sur le processus tumoral. Donc c'est des choses qu'Olivier Delattre a présentées dans son talk, mais on va essayer d'aller un petit peu vite pour vous donner quelques notions. Donc le processus tumoral, c'est quoi? C'est en fait une altération génétique, mutation ou autre qui arrive dans une cellule. Alors pourquoi elle arrive? C'est des notions qui seront discutées dans la table ronde cause et origine. Et cette altération va modifier, va transformer un peu cette cellule qui est normalement normale, formant une lésion précancéreuse. Et ces cellules vont se mettre à se diviser, et cette division va former une masse de cellules. Mais le système est un petit peu plus complexe que ça, parce que toutes les cellules cancéreuses ne sont pas toutes identiques. En fait, on a une hétérogénéité au sein de la tumeur. On a entendu notamment par Didier, on peut toucher cette hétérogénéité par Didier, mais il n'en a pas parlé exactement comme ça. Mais il faut avoir cette notion que toutes les cellules tumorales ne sont pas identiques au sein d'une tumeur, elles sont un petit peu différentes. Et le système est encore plus complexe que ça, c'est que les cancers sont des écosystèmes avec des cellules tumorales et des cellules normales qui dialoguent entre elles. Et ce dialogue entre ces cellules, c'est vraiment important. On va avoir par exemple des vaisseaux sanguins, mais aussi beaucoup de cellules immunitaires. Et on sait aujourd'hui que l'importance de ces cellules immunitaires dans la thérapie, notamment en raison des immunothérapies, et c'est quelque chose qu'on évoquera dans la session 4. Donc on va venir maintenant vraiment à la notion de cellule d'origine. Je pense que vous l'avez compris et Olivier Delattre a déjà un petit peu évoqué cette question. Un bref rappel, donc qu'est-ce que c'est en fait la recherche de la cellule d'origine? C'est en fait rechercher dans quelle cellule la première altération génétique va arriver. Et en fait, comme je vous l'ai dit, les tissus sont composés de différents types cellulaires en général, qui sont représentés ici en vert, en bleu ou en jaune. Et en fait, l'idée, c'est de savoir d'identifier où a eu l'altération dans ces cellules bleues, vertes, jaunes. Et c'est ça qui est recherché aujourd'hui. Et ça a une importance également parce qu'on pense, au moins dans de nombreux cas, mais pas dans tous les cas, que la cellule cancéreuse qu'on peut voir en bas va garder une certaine marque de cette cellule d'origine. C'est-à-dire qu'une lésion qui va arriver dans une cellule bleue va donner plutôt des cancers de type bleu, alors que des lésions qui vont arriver dans ces cellules de type jaune, on aura plutôt des cancers de type jaune. Donc, voilà. Je vais revenir à la diapo. Donc, en fait, on ne peut pas revenir en arrière, c'est ça? Ah si, voilà. Donc, en fait, c'est des choses qui sont importantes parce que Thomas vous a... parce qu'une même lésion génétique qui va arriver dans une cellule d'origine différente peut donner naissance à des cancers différents et c'est ce que Thomas vous a très très bien illustré. OK. Quel est l'intérêt de la recherche de la cellule d'origine? Bon, il y a déjà un intérêt cognitif, c'est-à-dire identifier d'où provient le cancer, mais c'est aussi, ça nous permet de développer des meilleurs modèles expérimentaux qui vont mieux mimer le processus qu'on va retrouver chez l'homme. Alors les modèles expérimentaux peuvent être des modèles animaux ou autres, et ça, dans la session 2, on va aborder ces modèles expérimentaux. Ça permet aussi de bien comprendre les premières phases où le cancer s'initie, c'est-à-dire comprendre un tout petit peu comment cette cellule commence à se modifier, à se transformer quand elle a reçu ce hit, et comment on va avoir une progression vers un cancer complet. Il y a également quelque chose qui est très essentiel parce qu'elle permet de bien comprendre les différences qu'on va avoir entre la cellule d'origine et la cellule cancéreuse. Et donc, comme ça, en identifiant ces différences, on va pouvoir mieux l'attaquer d'un point de vue thérapeutique. Et également, ça a un intérêt par rapport à la toxicité des traitements, que vous savez est important, puisque ça nous permet de tester, par exemple, la toxicité d'un traitement sur la cellule cancéreuse et sur sa cellule d'origine. Donc maintenant, je vais laisser la parole à Élise pour continuer un petit peu.
Donc, il nous a semblé important de rappeler quelques informations générales. Tout d'abord, des chiffres. Les cancers pédiatriques concernent 2840 enfants et adolescents chaque année. Ce chiffre est en constante augmentation. Ça représente à peu près un enfant sur 450 âgés de moins de 15 ans tous les ans. C'est d'ailleurs la première cause de décès des enfants de plus de 1 an par maladie. Ces cancers ont un taux de survie qui avoisine les 80%, mais ce chiffre masque de grandes disparités entre les cancers tout d'abord, notamment entre les rétinoblastomes dont la survie à 5 ans est de 98% et le DIPG qui a une survie à 5 ans inférieure à 1%. Une disparité également au sein des mêmes cancers, notamment avec la notion des tumeurs à haut risque qui sera développée dans la session 3 avec l'intérêt de la génomique. Et enfin, ce taux de survie masque une qualité de vie qui est souvent altérée pour les survivants par des séquelles des traitements qui concerneront 4 enfants guéris sur 5 qui présenteront des séquelles à long terme. Donc on comprend bien que les enjeux de la recherche, eh bien, ils sont doubles. Guérir plus d'enfants en identifiant notamment les cancers de mauvais pronostic ou à haut risque. Et puis, ils sont aussi de guérir mieux, avec moins de séquelles. Et pour ça, il faut connaître les particularités de ces cancers pédiatriques. Celio.
Ok, donc...
Pardon, non, c'est moi encore. Les cancers de l'enfant diffèrent de ceux des adultes. Effectivement, un certain nombre de constats sont que, par exemple, les taux de mutation à l'intérieur des cellules cancéreuses pédiatriques sont très inférieurs aux taux de mutation des cellules cancéreuses adultes, si on regarde la courbe de gauche pédiatrique et la courbe de droite adulte. On constate également que les diagnostics de ces cancers sont limités dans une fenêtre de temps très définie. Si on prend la courbe du rétinoblastome, qui est la courbe violet le plus claire, au-delà de la 7e année, on ne retrouve plus de diagnostic de méduloblastome chez les enfants. Enfin, le spectre de ces cancers est très différent de ceux de l'adulte, avec principalement, pour 50% des enfants atteints de cancer, des leucémies, des tumeurs cérébrales et des lymphomes. Alors, pourquoi et comment peut-on expliquer, Celio, ces différences ?
Donc, on va revenir. Donc, bien évidemment, on va revenir à cette cellule d'origine qui est pour nous quelque chose d'important. Je n'arrive pas à revenir. Voilà. Mais en fait, déjà, ce qu'il faut comprendre, c'est que l'environnement pédiatrique est très différent de l'environnement de l'adulte. On a des tissus qui sont en développement. Et donc, dans la recherche sur les tumeurs pédiatriques, il est très important de s'intéresser à la biologie du développement. Alors, qu'est-ce que ça signifie, un tissu en développement ? C'est un tissu qui est en croissance, c'est-à-dire qu'un tissu qui va prendre plus de place, qui a besoin de grossir. Et pour grossir, on va avoir des cellules qui sont en division, en prolifération ou en multiplication, peu importe comment on les appelle. Et on a aussi beaucoup de cellules immatures, et c'est quelque chose dont a parlé Richard Gilbertson au sujet de ce blocage de maturation. Alors que dans un tissu adulte, on aura un tissu qui est mature et beaucoup moins en prolifération. Donc, pour vous illustrer un peu, je voulais vous donner une idée de ce que la différence entre un tissu en développement et un tissu adulte. On peut voir ici, c'est un exemple du cervelet dans lequel se développent les méduloblastomes sur laquelle mon équipe travaille. On peut voir dans la partie gauche, dans l'encadré en bleu, un cervelet complètement mature. Et toutes les étapes précédentes, en fait, sont des étapes de développement. Donc, vous pouvez très bien voir que cette structure est très petite au début, elle va commencer à grossir. Elle ne fait pas que grossir, elle se modifie dans sa forme, donc ce qui implique beaucoup de mouvements, de migration ou de choses comme ça. Et donc, on peut facilement comprendre que des mutations qui arrivent au cours du développement vont avoir seront très différentes de mutations qui arriveront au cours de la vie adulte. Donc c'était exactement ce que je vous schématisais ici. Vous imaginez bien que même dans une cellule bleue claire, une mutation dans une cellule bleue claire n'aura pas les mêmes conséquences que dans une cellule bleu foncé, pour schématiser. Donc en fait, ces spécificités des cancers pédiatriques, le fait qu'elles arrivent dans des tissus en développement explique beaucoup de choses, notamment la toxicité importante des traitements, parce que évidemment on a des tissus immatures et que les traitements vont avoir une toxicité importante sur ces tissus-là. On va avoir des taux de mutation plus faibles parce que en fait, les mécanismes pour entrer dans les processus cancéreux vont être différents. On va, au lieu de réactiver la prolifération, on va certainement devoir plus bloquer la maturation de ces tissus. Voilà, et ça nécessite certainement un peu moins de d'événements oncogéniques pour y arriver, d'événements cancéreux pour y arriver. Donc, la fenêtre de temps dans laquelle on dit diagnostic qui correspond à un temps défini aussi s'explique facilement. Si on n'a plus la cellule qui est en prolifération, eh ben, le cancer ne peut plus se développer. Donc, on n'a plus de cancer à un certain nombre de temps quand la cellule n'est plus là dans le tissu adulte. Et le spectre de cancer différent est également lié en fait à cette susceptibilité de la cellule d'origine. Donc voilà. Donc je vais vous faire un petit résumé très rapide des talks qui ont été faits. Donc Thomas, qui vous a présenté ici, il a montré en fait que quand il introduisait, il s'intéresse à une leucémie aiguë mégakardioblastique pédiatrique, et qui est principalement dû à un gène de fusion qu'on appelle ETO2-GLIS2, et quand il introduit cette mutation ETO2-GLIS2 dans des cellules souches hématopoïétiques 2 fois fétales, il produit ce type de leucémie qui est vraiment très agressif, mais quand il introduit cette même mutation dans des cellules souches hématopoïétiques de l'adulte, il va former un autre type de leucémie avec une identité différente et dont le développement sera lent. Donc il illustre très bien comment la cellule d'origine a une très grande importance sur l'agressivité de la tumeur et sur son identité. Charles Mulligan, lui, s'est intéressé en fait à des leucémies qu'on appelle, dont l'identité est ambiguë. Alors, qu'est-ce qu'on appelle identité ambiguë? Je ne sais pas si vous avez très bien compris cette notion, mais comme je vous l'ai dit, une mutation, pour être très schématique, ce n'est pas complètement vrai, mais une mutation qui va arriver dans une cellule bleue va former plutôt des tumeurs de type bleu. Une mutation arrivant dans des cellules de type jaune va plutôt donner des tumeurs de type jaune. Et bien, en fait, ces identités ambiguës, on ne sait pas très bien si elles sont jaunes ou bleues. Et en fait, il a essayé de comprendre il a essayé d'identifier les altérations qui étaient présentes dans ces leucémies ambiguës, qui sont un peu de couleur problématique, un petit mélange des deux, et il a également cherché la cellule d'origine de ces leucémies. Alors, j'illustre un autre travail qu'a fait Richard Gilbertson, parce que je pense qu'il est très intéressant sur la cellule d'origine. Richard Gilbertson, dans des travaux précédents, pas le talk qu'il a présenté aujourd'hui, mais des travaux précédents, avait par exemple montré qu'on peut introduire des mutations qui vont toucher la voie sonic hedgehog. On va former des tumeurs de type bleu quand on introduit cette mutation dans les cellules de type bleu. Quand on introduit une mutation de type Wnt dans des cellules jaunes, on va former des tumeurs de type jaune. Mais ce qu'il montrait, c'est que si on introduisait une altération touchant la voie Wnt, WNT, dans des cellules de type bleu, et bien cette fois-là, on ne formait pas de tumeurs, alors que dans les cellules de type jaune, on formait des tumeurs, et le reverse était vrai pour Sonic Hedgehog. Et ce qu'il a montré, qui est très intéressant, c'est que si maintenant on introduit 2 mutations, un peu comme le fait Didier dans ses modèles, en fait, il est capable maintenant dans ces cellules de type bleu, avec une mutation de type Wnt, WNT, de former des tumeurs, et le reverse est vrai pour les mutations de type sonic dans ces cellules de type jaune. Et donc ça vous dit que, en fait, au départ, il y a une combinaison assez stricte entre une altération donnée et une cellule d'origine, mais que cette combinaison stricte peut être modifiée en fait par des mutations d'autres gènes qui va permettre une plus grande susceptibilité de la cellule d'origine au cancer. Enfin, Didier vous a présenté en fait des choses qui se passent sur le sarcome d'Ewing. Donc, comme il vous l'a dit, en fait, le sarcome d'Ewing est dû également à un gène de fusion qui va accoler 2 gènes, le gène EWS et le gène FLI. Et ce qu'il montre, c'est quand on introduit une nouvelle mutation, une mutation supplémentaire dans le gène STAC2, on va modifier un peu la caractéristique de ce cancer en lui conférant un caractère beaucoup plus agressif et métastatique, et qu'il a aussi mis en évidence les mécanismes qui étaient responsables de ça, qui sont assez surprenants, puisqu'il diminue en fait l'activité globalement de la première altération EWS-FLI. Donc voilà, je crois que maintenant on peut commencer la discussion. Donc on attend vos questions dans le chat. Donc une des premières questions qui touche ton talk, et notamment une des réponses que tu as apportées, Didier, c'est quelle est l'incidence du sarcome d'Ewing dans les différents pays? Car si la théorie dont vous avez parlé est juste, donc la théorie sur les mutations, sur je n'arrive pas à trouver le mot, mais tu vas le trouver, cette incidence internationale devrait être constante.
Alors, l'incidence est vraiment dépendante en fait du fond génétique, en effet. Et l'incidence est 10 fois plus importante dans une origine, on va dire, caucasienne qu'une origine africaine. Et pour l'origine asiatique, c'est environ 5 fois inférieur. Après,
Donc en fait, en Afrique, il y a beaucoup moins de sarcome d'Ewing que...
Alors, en Afrique, il y a certainement beaucoup moins de sarcome d'Ewing. Après, comment est-ce qu'ils sont vraiment répertoriés en Afrique, c'est peut-être aussi la question. Mais en tout cas, on sait que par exemple aux États-Unis, et ça c'est des études qui sont principalement faites en effet aux États-Unis, parce que l'origine ethnique en Europe, on sait bien que c'est une question qui est plus... Enfin, qu'on ne peut pas poser. Et du coup, on voit aux États-Unis, en effet, que l'incidence dans la population caucasienne est plus importante.
D'accord. Peut-être,
oui,
Élise,
j'allais dire, on peut peut-être rappeler aussi en 2 mots ce qu'est le sarcome d'Ewing, et puis surtout, maintenant que vous avez identifié cette altération stade 2 qui est responsable de cancers plus agressifs, qu'est-ce que tu peux espérer comme implication thérapeutique à tes recherches?
Oui, question très intéressante. Donc, juste pour rappeler, en effet, le sarcome d'Ewing, c'est une tumeur osseuse, principalement osseuse, 90% des patients ont cette tumeur à proximité d'os, plutôt les grands os. Maintenant, du point de vue thérapeutique, on sait qu'il y a, en fait, la molécule miracle serait celle qui, comme l'a introduit Olivier, va cibler cette translocation. Parce qu'aujourd'hui, on a des moyens génétiques d'inhiber la translocation. On voit vraiment que si on arrive suffisamment à l'inhiber, on arrive à tuer ces cellules. Et il y a suffisamment d'autres exemples maintenant dans la littérature pour dire quand on attaque la translocation, par exemple comme l'a montré Olivier avec les NTRK, ça fonctionne. Malheureusement, on est dans un cas de figure où le sarcome d'Ewing, ces protéines, elles sont à l'intérieur du noyau. Et aujourd'hui, on n'a pas vraiment les outils pour fabriquer les médicaments qui vont cibler ces facteurs de transcription. Alors, j'irai encore une étape plus loin dans le travail qu'on a fait là sur stade 2. Alors, stade 2, cette mutation fait qu'en fait, on perd l'expression de stade 2. Du coup, ça devient très compliqué de cibler quelque chose qui n'est pas exprimé. Alors, là, de nouveau, là, il y a des alternatives. Et malheureusement, de nouveau, elles ne sont pas facilement ciblables. C'est que je vous ai parlé un petit peu de stade 1. Et on sait que dans la cellule qui est mutée stade 2, il n'y a plus de stade 2. Donc, c'est stade 1 qui prend le rôle de stade 1 et de stade 2. Donc, maintenant, il y a plusieurs groupes qui ont pu montrer que lorsqu'on inhibe stade 1, dans un contexte où il n'y a plus stade 2, la cellule meurt. Et c'est une approche thérapeutique extrêmement intéressante. C'est un concept qu'on appelle un peu compliqué de létalité synthétique. Mais en fait, on profite de l'avantage qu'il y a la mutation dans la tumeur et que dans les cellules normales, il n'y a pas cette mutation, donc pas perte de stade 2. Et que si maintenant on arrive à inhiber stade 1, eh bien, les cellules tumorales meurent alors que les cellules normales ne mourraient pas. De nouveau, le problème...
Vous avez pu montrer que c'est effectivement le cas dans les sarcomes d'Ewing ?
Dans nos modèles et dans différentes publications, c'est vraiment ce qui ressort. Et là, de nouveau, malheureusement, stade 1 est une protéine de nouveau que je vous montrais, va se lier à l'ADN, à la chromatine. Et là, de nouveau, on n'a pas les outils pour cibler ça. Donc vraiment, une des approches et un des buts du laboratoire que je développe maintenant, c'est d'identifier quelles sont les autres protéines qui pourraient agir comme stade 1, qu'on pourrait cibler, qui permettraient d'apporter une thérapie ciblée pour ces sarcomes d'Ewing particulièrement agressifs.
Ouais, donc juste pour rajouter un mot, je pense que ce que les gens doivent bien comprendre, c'est que effectivement, ces cellules-là ont une faille qui leur est très spécifique, qu'on ne retrouvera pas dans les cellules normales et qu'on peut identifier des traitements qui seraient très peu toxiques pour les cellules normales et très efficaces sur les cellules mutées. On va peut-être passer un petit peu à toi. Thomas, je crois que tu avais quelques questions.
Oui, on a entendu parler de la notion cellule d'origine depuis le début des présentations. Tu nous parles de la notion de cellule souche. Est-ce que tu peux nous distinguer un petit peu, nous préciser un petit peu la différence ? La cellule souche est une cellule d'origine ?
La cellule souche peut être une cellule d'origine. La difficulté, c'est qu'est-ce qu'on met dans le concept de cellule souche. Alors en fait, on entend beaucoup parler de cellules souches, par exemple des cellules embryoniques stem ou des cellules pluripotentes, qui sont capables de reconstituer en fait tout un embryon et tout un individu par la suite. Les cellules souches dont j'ai parlé dans la présentation sont des cellules souches tissulaires d'un tissu donné et qui sont capables de reconstituer les différents lignées qui sont retrouvés dans ce tissu spécifiquement et pas, par exemple, les cellules souches hématopoïétiques ne sont pas capables de redonner des cellules de l'intestin. Donc, effectivement, l'objectif, en fait, qui a été bien résumé par Celio, c'est de comprendre, tout d'abord, quelles sont les altérations qui sont à l'origine de la progression tumorale. Et comme Didier SURDEZ l'a expliqué, l'objectif des modèles, c'est de comprendre quelles sont ces altérations qui sont importantes, quelles sont les combinaisons. Et ensuite, l'autre objectif, c'est de comprendre dans quel contexte elles doivent survenir pour entraîner un développement tumoral. Et les résultats que nous avons pour les leucémies de l'enfant, cet exemple particulier, ça peut ne pas s'appliquer à toutes les leucémies. C'est ça qu'il faut aussi comprendre, c'est qu'en fait, il y a un certain nombre de possibilités qui ne sont pas toujours les mêmes. Dans le cas de ces leucémies à fusion et taux de Gliese 2, il semble très important que la cellule d'origine soit une cellule souche hématopoïétique qui soit capable de reconstituer un certain nombre de lignées hématopoïétiques. Donc ça, c'est la première capacité. Et la deuxième, c'est qu'elle soit dans un environnement plutôt développemental et pas adulte. Sachant qu'une cellule fœtale, une cellule souche fœtale, les cellules souches hématopoïétiques fœtales, normale est capable de redonner tous les lignages et une cellule souche adulte est également capable de redonner toutes les lignées hématopoïétiques. Mais qu'ensuite, il y a des différences d'état, par exemple l'état de prolifération, qui donne des différences de propriétés biologiques. Je ne sais pas si j'ai entièrement répondu à la question.
On va retourner au chat. Donc on va finir, on va prendre le temps pour 2 dernières questions parce que je trouve que c'est intéressant d'avoir des questions du chat. Je vais une pour toi et une pour Didier. Donc, connaît-on les causes des leucémies aiguës lymphoblastiques? Question difficile, je crois.
C'est une question large. Comme vous l'avez en partie entendu, il y a un grand panel de différentes leucémies. Et l'origine est probablement différente d'une leucémie à l'autre et potentiellement d'un individu à l'autre. Vous avez vu que certaines leucémies ont plusieurs oncogènes, donc c'est un processus multi-étapes. Et chaque événement s'accumule dans une cellule. Alors on sait qu'il y a des choses qui prédisposent au développement de leucémie. Par exemple, les irradiations aiguës, fortes. Il y a un certain nombre d'autres expositions dont on ne connaît pas véritablement l'effet et qu'on suspecte comme étant prédisposantes au développement de leucémie. Donc la réponse, elle ne peut pas être simple en une phrase pour toutes les leucémies aiguës lymphoïdes, ni même myéloïdes. Et il faut continuer à faire des associations à la fois épidémiologiques et moléculaires pour bien comprendre le lien entre les deux, entre les expositions et les conséquences sur les cellules.
Je pense que c'est des choses, de toute façon, sur lesquelles vous reviendrez pendant la table ronde cause et origine du cancer. Hein.
Absolument.
Exactement. Donc, c'est quand? C'est demain, c'est ça?
Vendredi.
Vendredi. Donc, vendredi, ces choses-là seront discutées. Une dernière question pour toi, Didier, même si on est un petit peu en retard. C'est, pensez-vous qu'il y a une influence de l'environnement cellulaire sur l'apparition de la mutation STAC2?
Alors, sur la mutation STAC2, c'est une excellente question à laquelle on n'a pas encore complètement la réponse. Donc, c'est un travail qu'on est en train de faire également en collaboration avec l'équipe d'Erika Brunet, où de nouveau, on essaie de prendre la cellule d'origine. Alors, dans le sarcome d'Ewing, c'est une cellule souche mésenchymateuse, et par rapport, on va dire, aux leucémies, le concept de cellule souche ou la caractérisation de ces cellules souches est nettement moins bien défini. Et dans ce contexte-là, on a essayé de prendre des cellules souches mésenchymateuses de patients qui étaient atteints de sarcome d'Ewing, de récapituler en fait cette translocation. Et ce qu'on a pu observer, c'est que notamment quand on ajoutait cette mutation STAC2, on favorisait finalement la pousse de cette tumeur. Donc, je ne réponds pas complètement à la question, mais aujourd'hui, le seul élément de réponse pour dire comment intervient STAC2 dans ce processus, c'est de dire qu'il semble que STAC2 favorise également la prolifération des cellules.
En fait, je pense que c'était plutôt sur pourquoi elle arrive cette mutation, tu vois, et quels étaient les facteurs qui pouvaient conduire à cette mutation.
Pourquoi est-ce qu'elle... on voit quand même qu'il y a un processus sélectif, notamment chez les patients. Là, de nouveau, on n'a pas la réponse, mais notamment quand on regarde des tumeurs au diagnostic et des tumeurs à la rechute, on a un certain nombre d'exemples où on voit qu'il y a une sous-population de la tumeur qui a une mutation STAC2, et on voit qu'après les chimiothérapies, après les irradiations, dans un certain nombre d'exemples, on voit que c'est finalement la cellule avec la mutation STAC2 qui a résisté au traitement. Donc il se peut qu'il y ait aussi d'autres mutations, mais que finalement les autres mutations ne confèrent pas cet avantage de résistance aux thérapies qui sont actuellement utilisées.
Ok, je pense qu'on va conclure ici.
Oui,
Élise, je te laisse finir.
On va faire une courte pause. On se retrouve à 18h10 pour la conférence de Michel Monge de l'Université Stanford intitulée « De la cognition au cancer » sur le rôle des interactions neuronales dans la physiologie et la pathologie. Merci à tous les trois, merci à tous nos intervenants et restez connectés.